Good afternoon. If I could have your attention. Good afternoon. Thank you. Good afternoon. I am Rachel Bronson, President and CEO of the Bulletin of the Atomic Scientists, and we're delighted to welcome those in the room and those who are now joining us via live stream. This conference marks the midpoint of a year-long task force that the Bulletin has convened, focused on creating the framework for tomorrow's pathogen research. The task force has met 4 times leading up to this conference. Our goal over the next day and a half is to begin sharing some of the insights generated by our previous discussions and now including a wider audience into those discussions. The comments and perspectives that we receive here in Geneva will inform our final report. Following this conference, the task force will continue to meet and integrate what we heard today and tomorrow into a set of final recommendations that we expect to publish later this fall. The task force, as those in the room know, is chaired by Ravi Gupta, Amina Garib Fakim, Shahid Jamil, and David Relman, whom we will hear from shortly. It is directed by Jesse Bloom and Philippa Lensos, and we're supported by 2 excellent rapporteurs, Myra Amaneros and Becca Earnhardt. Bios for them and all of our speakers can be found on the Bulletin's website at thebulletin.org for those on the live stream, and for those in the room on the QR code on the back of your name cards. The Bulletin, as many of you know, is more than 75 years old. We are best known for our nuclear work, but over the last several decades, we have focused increasingly on climate change and new disruptive technologies such as genetic engineering and those included in new biosciences research. What connects the issues we cover is a focus on technologies that have the potential to end life on Earth as we know it. We believe that we can control what we create. In fact, we have a responsibility to do so. Most technologies are dual use, with the possibility, nay, the probability, of bringing great triumphs and great tribulations alike. Understanding this and constructing effective governance regimes is not easy, but it is the only path to ensuring a healthy, thriving, and sustainable future. The Bulletin's founders understood this instinctively in the nuclear space, but in other spaces as well. Since our creation, the Bulletin has also addressed advancements in biological sciences, first through the lens of biosecurity, and more recently through the lens of biosafety. That is the focus of our discussions today and tomorrow. We are delighted to be joined by a cross-section of scientists, social scientists, policymakers, and institutional leaders. Those in the room today are an extraordinary group, and we know we are in for a set of fascinating discussions. For those on the live stream, we'll be collecting your questions and comments via Twitter at the hashtag Pathogens Project. For those on Twitter and in the room, please remember to keep all comments respectful. By attending this conference in person or via live stream, we expect that everyone will conduct themselves in a manner that fosters positive, productive environment. This includes refraining from using inappropriate language and physical gestures and allowing for the free flow of information and dialogue. We are extraordinarily grateful to our funders, with whom this meeting and the task force would not be possible. In addition to very generous anonymous donors, we are grateful to the Gurley Family Charitable Fund, Robert and Eleanor Myers, and Keith D. and Arlene M. Bronstein Foundation. Let me now turn the podium over to David Relman, who will get us started. David is the Thomas C. and Joan M. Meridian Professor in Medicine and a Professor of Microbiology and Immunology at Stanford University and the Chief of Infectious Disease at the Veterans Affairs Palo Alto Healthcare System. He is also a co-chair of the Bulletin's Pathogens project and a member of the Bulletin Science and Security Board. David, over to you.
Well, let me start by making clear that, to me, this is a great honor and a pleasure to be here, and I'm particularly excited that we're able to be here in person, and I'm able to meet a number of you that I have not been able to put an analog face to the name. So I'm grateful for that. But I also am quite aware that it's a daunting task to provide comments that can be useful at the opening of a meeting with a bunch of really smart people who truly understand all of the issues that are at play here during this meeting. So I'm going to try to focus on both some personal comments as well as perhaps some calls for issues that might deserve special attention during the next 2 days. And I'm going to comment in particular about recent history, about responsibilities, about needs, and then some suggestions about areas where we might focus over the next day or so. I need not remind all of you that we are living in the midst of a revolution in the life sciences, and one that has increasingly made clear both immense promise and as well risks. And that's where, of course, the tension and the challenge lies in trying to resolve and reconcile those two aspects of this emerging area of science. In many ways, the first meeting of this group took place in Asilomar, California, in 1975, February. But it was a very different world, because in February of 1975, although many had just realized the beginning of a capability that had not ever been imagined, really, that is, of recombinant DNA, there were only 140 people who were present and who essentially comprised the world of people who were capable of engaging in this new science. And so it's perhaps humbling to think that just 10 years later, 1985, was the first description of an example of this new science that has particular relevance to us today, of a form that's called reverse genetics, the ability to start with a sequence and convert it to the biological entity that bore that genetic sequence. In other words, to create a virus, for example, from a sequence that had whatever features one might have desired or designed into that starting template. But in 1985, of course, there were very few practitioners who could undertake such work. It was costly, it was laborious, and there were lots of technical challenges. The sequences had plenty of errors, they still do, but it was difficult to recognize them. It was also problematic to synthesize the sequences, assemble them, and know what kinds of accessory functions had to be provided at the same time in order to boot up this virus. And yet, 1985 was polio, 1994, rabies, 1996, influenza, and so on. And what became clear was that all of this was not only possible, but it was increasingly feasible and simpler to do, made possible by a great deal of science that sought to use this technology to understand the living world better and perhaps to create interventions that would better all of us. By 2001, there were examples in the scientific literature now of experiments that followed on this theme, but started to prompt the realization that there might be some risks, some attendant risks. 2001 was the famous, or maybe now infamous, ectromelia virus IL-4 experiment. And through the subsequent decade or 12 years, increasing numbers of these experiments became known, and with them, increasing awareness of this other side, this other possibility, from the work that had been undertaken again for, the clear betterment of us all. And by 2012, we now had, at least in my estimation, an experiment and an example of something that I think perhaps resonated with a large enough number of all of us, scientists, and the rest of the public to now raise questions at a level that almost demanded some attention. And this was the work of H5N1 influenza and the effort to understand the possibility of mammalian transmissibility. But at this point, what I think becomes clear is that there are now questions being raised by the rest of our colleagues in society around the world about the appropriateness of these experiments and raising questions about the trust that we had assumed we had with all of our societal members who are funding the work and hoping to receive nothing but benefit. And then became clear that we needed to pause and have a more deliberate conversation. And that's, in the United States at least, the moratorium that the US government undertook on so-called gain-of-function studies between 2014 and 2017. And what came of that, I think, was the realization that not only were there a need, but a means to begin to have conversations that, again, were more deliberate and focused on how do we assess risk and benefit. And I know a lot of you were involved in the efforts in those years, not only in the United States, but elsewhere. This is still an ongoing process, as you well know, and it prompts, again, a meeting of this sort. But this little brief history related to this meeting, I think, also deserves mention of the pandemic that is still ongoing, that we have all suffered through in various forms over the last 3 years. To me, there are a number of important lessons, again, for perhaps consideration at this meeting. It's not just the consequences for the public health, which have been substantial and painful, but it's the lessons about transmissibility, the unusual importance of transmissibility, and how a fast-moving entity like a virus can outrun even the best defenses that we might have already in place, for example. It also teaches us that, I think, to Rachel's comments, it's we, the human species, that have increasing degrees of control over events of this sort, because regardless of how this pandemic began, I think we can agree that in some general form, humans facilitated an encounter between ourselves, the species, and this virus. It happened somewhere, likely because of some forms of human activity, trade, commerce, laboratory research, any of a number of activities that probably enhance the likelihoods of this encounter in some place at some point. And that's another important lesson to keep in mind. And finally, the unbelievable attention now that the public has on the issues before us, and I think the responsibility that we have to address them in a thoughtful manner. Now, there are a lot of pressing needs that we have in science and in public health, and I don't want any of that to be lost. Attention has returned to work of the sort that the the original Global Virome Project proposed. That is, to understand better the world around us. What is it that might already exist that we need to understand better if we're going to have any chance of anticipating and preempting the potential harm? And there's a lot of attention now on how we go about doing that. And the bottom line is that there's an incredible need for a balance. It's going to be a balance between the imperative of understanding our world and yet doing so in a manner that does not, by its very process, either exacerbate risk or create new ones. We have to be mindful of that now because we have arrived at the point where we have these capabilities to undertake work that does have those potential consequences. Now, there are issues of safety as well as security, and we will be exploring those in much greater detail. I would only urge that we think carefully about what we know and don't know about assessing the possible response to concerns over security and safety. As an example, I'm impressed with the fact that as a former biosafety panel manager at my university, we don't fully understand all of the work that's being done and the possible consequences. And if you want any documentation of that, turn to nothing more than the work of Alison Young in USA Today, this investigative reporter who, as you probably well know, issued a lot of Freedom of Information Act requests asking for details about what panels like mine knew or perhaps should have known and didn't. And what, of course, becomes clear is much more concern or possible events of concern than, of course, we were aware and perhaps work that we should have been aware of. So some humility and honesty is what I'm suggesting. And a willingness to take responsibility. I want to call out two papers that were published around the turn of the last century, meaning 1999, the year 2000, by a physicist named Joseph Rotblat. Rotblat was a Polish physicist trained in the 1920s in Warsaw, and then undertook work which took him then to England, where he was among the first to realize the release of neutrons upon fission and the possibility of a chain reaction that might unleash enormous, previously undreamt amounts of power and energy. The potential of an atom bomb. And accordingly, he and his colleague in Liverpool were called to Los Alamos to join this Manhattan Project. This is now 1943. Rotblat writes in more recent times about his thinking, about what might have motivated someone to join such a project knowing the potential consequences of it. And he writes that it was the fear that others might undertake this work for clearly harmful purposes, thinking of Adolf Hitler and the early years of World War II. But when 1 year later, in 1944, Rotblat learned in Los Alamos that the Germans had abandoned their atom bomb project, he became very, very disillusioned, and he left. He abandoned the Manhattan Project and moved back to England, where he began a second career in health physics, so the use of radiation for health purposes. And he then went on to work tirelessly to alert the world of the potential hazards of the very discoveries that he had helped contribute to. He won a Nobel Prize, Peace Prize, in 1995, and did a number of things like create the Potsdam Conferences. But Rotblat wrote in 1999 a piece called A Hippocratic Oath for Scientists. This is the November 19th issue. And I just want to read some of the words from that paper. He wrote, "Science has become a dominant element in our lives. It has enormously improved the quality of life. It has also created great perils, threatening the very existence of the human species. Scientists can no longer claim that their work has nothing to do with the welfare of the individual or with state policies. However, many scientists still cling to an ivory tower mentality founded on precepts such as science should be done for its own sake, or science is neutral, or science cannot be blamed for its misapplication. Their logic," he wrote, "rests on the distinction between pure and applied science. It is only the application of science that can be harmful, they allege. As for pure science, they say that the scientist's only obligation is to make the results of research known to the public. What the public does with them is its business, not that of the scientist. This amoral attitude is, in my opinion, actually immoral because it eschews personal responsibility for the likely consequences of one's actions." And I think when he wrote this in 1999, many thought this the unusual province of the physics world, the physicists, and the world that they had now they had inherited from this history in the 1930s and 40s. What I think I and maybe perhaps many of you would suggest is that we too, the life scientists, have now arrived at a time when we too have to consider potentially existential dangers that deserve the same kind of consideration for personal responsibility that the physicists took on, or at least some, through admonitions like that of Rotblat. And I just want to add that with respect to responsibilities, remember that in our quest for benefit in science, we also have to be aware that there's very often unequal sharing of those benefits around the world. This is the issue of justice. And there's also a temporal uncoupling of risk and benefit, because if the work we publish today provides both, it's quite often the case that the risk is acquired now, but the benefit may take a bit more time. And that's this time disconnect that I want to just comment upon again briefly. So as far as the needs, I would say that there are three. And the first is that we consider a portfolio of possible mechanisms for addressing this conundrum of risk versus benefit. One analogy is, here we are, we're hurtling down a road or a would-be road that we perhaps hope to stay upon in some stable, responsible fashion. But in our haste, and because our vehicle doesn't always have great controls, and because we're distracted as drivers with our, not just our little devices, but our thoughts, perhaps we ought to consider guardrails. And guardrails are sort of, again, a metaphor for one kind of mechanism we might want to consider. And I'm going to comment upon that for just a few more minutes as well. But there are other possible mechanisms, and I don't want to preclude any of these, nor to suggest that any one will ever work alone. But another might be the analogy of the light post, providing greater clarity on how to stay on the road. And we might think about what is a light post in the life sciences world today. So with respect to guardrails, I'm referring to something that in history has been referred to as red lines or lines in the sand. It's a place past which one shall not proceed. It sometimes is used or thought of as an ultimatum or a threat. And these red lines, have been called upon, referred to over time in all kinds of varied, sometimes very vague ways, thereby creating doubts as to whether even the concept may work or is appropriate, especially in the context that I want to propose here in the life sciences. Red lines offer some benefits as guardrails. They serve as deterrents. They sometimes are used to refer to something called coercive diplomacy. They also reinforce norms, but they often don't work for a variety of reasons. First, because they require clear objectives. They require the clarity of circumstance. Exactly when is it that they are best used and for what purpose? They also require clarity of response. You have to be clear about what it is that you seek to prevent and how you know whether this thing occurs or not. And those in the world of game theory, people like Thomas Schelling, have said they only work if you have not only threats but commitments or promises of benefits, of positive incentives, should you adhere or follow these red lines. And then finally, you also need credibility. In other words, the capacity, the authority, the will, the interest to enforce them and act upon them. So in essence, red lines have to be carefully calibrated if they're to have any chance of working at all. And there are plenty of people who have written that basically they often don't work because those requirements simply are not met, nor can they be met under a number of circumstances. So I'm left uncertain as to whether they have a role. But I will say that we are, without them right now, at a point of lack of clarity. We are not clear as to whether there are experiments that we shall not pursue, that we should not undertake, and that we have a general consensus on this. And I think that's perhaps something that might be worth considering in the back of your mind as we talk over the next 2 days. Because again, if we simply hurtle down a road and hope for the best, by the time we discover the need for a response or some mitigating approach, we're playing catch-up. And yet, again, I'm not sure that red lines are necessarily practical. They certainly have concerns about the possible stifling of science or information hazards that can result. So some in political science have said, well, sometimes you don't use a red line, you use strategic ambiguity. And I think we're sort of, by default, using strategic ambiguity right now, again, with unclear likelihoods of success. Because you've got flexibility, but they may not be effective or credible at all. Which leads me to underscoring this problem of time disconnect. There's an interesting other person that I'm going to suggest you look at, although it's hard to find this book. The book is called Social Change, and it was written by a sociologist from Columbia University in New York in 1923, and his name is William Ogburn. And Ogburn coined this term cultural lag. And it arose because he wrote that in the 1920s, they were witnessing an unprecedented time of radical change in technology, like the world had never seen. And that it was leaving behind the social and cultural institutions that allowed everyone to adapt and accommodate these technological changes. And he talked about industry, industrial production, and factories, and yet the occupational health guidelines that were way behind the times. And he said, the thesis is, I'm reading from his book, the thesis is that the various parts of modern culture are not changing at the same rate. And he's referring to industry and education. Where one part of culture changes first through some discovery or invention, and occasions changes in some part of culture dependent upon it, there frequently is a delay in the changes occasioned in the dependent part. He's got a little bit tough to follow. But the bottom line is, you've got this lag, and it's a period of maladjustment, and we need to address it. And I'm taken by that because, in essence, that's what we've got right now. We've got science moving along so fast that by the time we realize that there are some issues at hand that need to be thought about and pondered, the science is already further ahead, and we haven't had time to have that kind of discussion that I hope we have here. So, again, think about how we might build into the future periodic moments for deliberate thought and discussion. Perhaps in a very deliberate fashion, this might be implemented. You know, taking the road analogy, these would be speed bumps. This would force the cars to just slow down for a minute so that we can catch up sociologically, psychologically. And then the last need I want to highlight before my last few comments is the need to take seriously the importance of information hazards. We, the larger we, have, in my view, ignored or paid insufficient attention to information hazards. When we think about biosafety, we're thinking about physical hazards. And yet, again, going back to those early developments that I highlighted in the history of a form of life sciences technology, the reverse genetics, have sequence, make virus. Make virus anywhere in the world that one has the means and the know-how to do so, which is increasing numbers of places. And so you can't simply say, the work began here, and here we have good biosafety, end of story. That's only the very beginning of a story that's going to be much more difficult, admittedly, but one that has to be sort of thought through. Okay, so some ideas of things that we can, again, think about and focus upon here. Let's think about who we are as a group. This is an amazing group of people. I don't think I've been at a meeting with the diversity of backgrounds, of expertise, of cultures, of worldviews to talk about these issues before, at least for me. And I would urge that we take advantage of that in every way possible. I would urge that we think about these moral responsibilities. The world is now looking and listening to us. And as I've said, the public expects that we start to assume responsibility and take proper steps. In not only our own view, but in the view of all the others who care, have to care, because again, the consequences of our action have repercussions for everyone else on this planet. It's not just those in the laboratory and our families. It's everyone else. Third, I suggest that we promote certain principles here that include transparency. That's how you build trust. That's how you have good, serious discussions that are objective and rigorous, and that we adopt the principle of accountability as well. Fourth, if nothing else, I would suggest that this meeting might help motivate and legitimize conversations about risks among other scientists who haven't perhaps had the chance to do so or to stop and to think about the importance of doing so. We ought to promote that as well. And then finally, we're going to talk about lots of complementary strategies. I've mentioned guardrails and speed bumps, and there are lots of perspectives on which to base different approaches. Bottom up, from the worker, the scientist, the person on the ground in the field, or top down from institutions and governments and so forth, organizations. They both have importance. I would suggest that none of this is either or, it's all yes and. Think about actors. There are individuals, there are institutions, etc. I'm not sure that we have properly identified all of those that are most important or will be most effective. And then finally, let's talk about process, how to be deliberate, representative. Representative of what? Who are the constituencies that would like to be here, didn't know they wanted to be here, but do? And then we should be, finally, self-critical. Let's not suggest that we have answers. I'm not sure that anyone here will, be in danger of doing so. But let's also just make sure that what we start here or continue here continues on afterwards and is an iterative, regular process of self-assessment. I will stop there. I'm looking forward to this meeting greatly, and I thank you for sort of indulging me in these comments. Thank you.
Thank you. This now changes us to the next portion of our sessions. You can exit out those doors and come around, and we'll reconvene in about 15 minutes in the main room. Thank you.
Excellencies, 1 month ago, we turned the calendar on a new year. But just days ago, another clock turned, the so-called doomsday clock.
A short time ago, an American airplane dropped one bomb on Hiroshima and destroyed its usefulness to the enemy. It is a harnessing of the basic power of the universe.
That symbolic clock was created 76 years ago by atomic scientists, including Albert Einstein. Year after year, experts have measured humanity's proximity to midnight. In other words, to self-destruction.
Negotiations were concluded in Moscow on a treaty to ban all nuclear tests in the atmosphere, in outer space, and underwater.
Mr. Gorbachev, tear down this wall.
Today, at 15:45 hours, India conducted 3 underground nuclear tests in the Pokhran range.
This means that the spores used in the attacks were taken from that specific flask, regrown, purified, dried, and loaded into the letters.
Events like COVID-19 can no longer be considered rare, once-in-a-century occurrences. The total number and diversity of infectious disease outbreaks has increased significantly over the last 40 years, with more than half caused by zoonotic diseases. That is, disease originating in animals and transmitted to humans. As such, zoonoses put the human population at significant risk for pandemics.
In 2023, they surveyed the state of the world and they came to a clear conclusion.
It is now 90 seconds to midnight.
Which means 19 seconds to total global catastrophe. Now, this is the closest the clock has ever stood to humanity's darkest hour, and closer than even during the height of the Cold War. In truth, the doomsday clock is a global alarm clock. We need to wake up and get to work.
Hello, everyone. Hopefully you can hear me. My name is Jesse Bloom, so after that very sobering video, I'm going to be leading this first session, which is biorisks in the future. So we're fortunate to have 4 excellent scientists on our panel today. I'm going to briefly introduce them and refer you to the online material for detailed biography. We have Volker Thiel, who's at University of Bern, Gustavo Palacios from Mount Sinai, Claire Jolly from University College London, and Ravi Gupta from University of Cambridge. And our goal on this panel is to discuss some of the scientific issues and sources of risk for sort of biological events in the future. So the first question, which I'll start off leading with Volker, is sort of what do you perceive as the biggest risks that we face going forward, both from zoonotic and biosafety, biosecurity perspectives?
So I think you already distinguished in your question between zoonosis and the other aspect is biosafety. Let's start with zoonosis. I think it's quite obvious that we essentially invade nature at the moment. We have much more contact to wildlife, and I think that's where there is considerable risk. Also the way how we deal with, let's say, animals mainly. You may know that the markets in China have been one place where at least it can be considered that virus can spread easily. So our behavior, how we deal with nature, how we deal with animals, how we live essentially our lifestyle even is maybe concerning zoonosis one of the most important way how we can maybe generate
and I think that's a good point. I mean, I think when we think of the markets, we think not only of wet markets like we've discussed in China, but for instance, we saw an H5N1 outbreak on a mink farm, in Europe, and I think there's lots of things like that are generating risks.
So the other thing when it comes to biosafety, biosecurity, let me maybe start with where I have not a big feeling that there is a big risk. I mean, risk is always the question, if something happens, what impact does it have and how likely it is to happen? And I think in many countries we have very good regulations on the work that is done with, let's say, these high pathogenic viruses or pathogens. These are countries, let's say, where people stick to the law, where you have regulations, where everything is also on the technical standard that you can be quite safe. It does not mean that nothing can happen, but I think here we are rather good. Where I have a little bit of fear or where I think there are issues is in areas where it's not regulated properly, where we maybe have research outside any regulation. So this is sort of maybe the candidate areas where we should do something.
Yeah, and what would you see as examples of that sort of research that's going on in areas that are outside of the standard regulation?
Well, those examples you can hardly name because they don't tell you I'm doing something outside of regulations, but you can imagine that those things can happen.
Yeah, no, those are very thoughtful answers. And I'd like to also pass that question to Gustavo, and maybe Gustavo, you could also elaborate a little, as people may or may not know. Gustavo's now sort of an academic scientist like all of us here, but prior to that, you were working at US AMRID, which is a different sort of more regulated or highly regulated environment doing research. I'm curious if that gives you any perspective on these issues.
Yes, I actually am going to mention and follow up with the word, one of the words that is regulations. The other word that David Relman used in his introduction was the strategic ambiguity and the fact of how, in terms of our analysis of the risk, we are considering always that there is a careful evaluation of the risk before we are taking an action. And it seems that one of the major risks that I am seeing now is that we have a very nonchalant approach to do research and on high containment pathogens or potential high containment pathogens. And so in the past, one of the ways to handle that was to ensure that national laboratories and government laboratories were involved in a lot of those types of research. And those are heavily regulated, slow, very bureaucratic, but there is a reason why they are like that. We have moved a lot of that research to academia that actually is is a lot less regulated. Maybe that is a problem that we need to consider. And the second aspect is, at least in the US, government scientists are not as prestigious positions as they had been in the past, and therefore there is a degradation of talent in some of those areas that maybe are not attracting, the pool of individuals that could sustain that type of work in a very regulated way and advancing the science that is needed to control these risks.
Yeah, no, that's a good point. And I think Gustavo makes a valuable point here in terms of a lot of research in academia, not having the same amount of vetting and review and regulation as research in some sort of government labs. On the flip side, and maybe as we go to Claire and Ravi, I mean, we have seen during the COVID pandemic, there've been a lot of academic scientists, both of you being examples, myself being another example, who because of sort of the flexibility and relatively lighter regulation in academia were able to quickly pivot from working in other areas to working on viruses like COVID-19. And so I'd also be curious to hear your perspectives on what you see as sort of these risks and, both from zoonotic and biosafety perspectives.
Thanks, Jesse. I think UCL would argue that we weren't more slack on our safety, which allowed us to work on SARS-CoV-2. I think the guys summed up a lot of the same points that I would have made. I guess there's two, in terms of zoonosis, absolutely increased interaction with animals, and I think that obviously involves intensive farming and wildlife trade, but also I think something that's quite important is obviously climate change. So increasing not only the frequency of our interactions with animals, but the types of interactions we have, particularly changes in vector distribution. So mosquitoes, ticks, for example, we're seeing in Europe increase in frequency, and tick and mosquito-borne infections that we haven't had in the past because of changes in ecology. So I think that is an important contributor to increasing zoonotic risk. In terms of sort of biosafety and biosecurity, I agree very much that in a lot of the world, particularly the UK and where most of us are working, everything is very heavily regulated, tightly controlled. Safety is paramount. I guess something that I've been thinking about over the past couple of years is field sampling. So people going into taking ecological samples or samples from animals, either intentionally seeking to understand what potential viruses and bacteria may be there, but additionally people sampling for other purposes, such as routine surveillance, or groups who may not be thinking about what might be in the samples that they're taking because it's not what they work on. They're not virologists, for example. So how are those samples taken? How are they stored? How are they processed and worked with for people who are not maybe as tuned into thinking about the risks of what you're not used to thinking about? So I think that, for me, is something that is an important consideration.
Yeah, I'd like to add, I think, to that the fact that unintentional exposure is probably quite a big risk. For example, if you're not working on a particular virus, but you happen to have a sample that has a different virus in it, this is I think a really important area where frameworks that are adopted globally are important. We do need international frameworks for processing animal samples, whether it's blood, tissue, or other products. So there's certainly something that can be done there, because it's fairly. But then the question will be, of course, if you identify a framework, which labs have the capacity to process samples within Category 3 containment, for example, or do you derogate everything to Category 2? So some of these will be pragmatic decisions, because it's going to be quite risky to generate a framework that very few people can adhere to, and that actually polarizes the world into those who have and those who don't have. So I think that is tricky, but absolutely essential. Just going to the question about zoonosis, I think that, yes, it's a huge problem. I can't see it getting any better through regulation. I mean, of course, many of these things are entrenched in society. We are, of course, becoming more intensive in the way we do everything. It's very hard to see how we will draw back from, man's quest to to generate capital and wealth. You know, so I think that the way to mitigate that, you know, it's ambitious, but I think it's something that maybe the US government is already thinking about doing, which is to categorize and document all, you know, as many different varieties or viruses that are out there, and then generate, you know, ranges of vaccines, for example, so that you have an array of vaccines against different groups of viruses, so that if there is a spillover event and we manage to detect it, that we can rapidly scale up vaccines, for example. So you could look at it from a mitigation point of view rather than how do we stop the source of that spillover, which I think is a much bigger, well, they're both equally large tasks, and maybe what we need to do is a bit of both.
Yeah, that's great. And to kind of play off that, because Ravi's sort of leading into my next question, which is sort of what can we do to mitigate these risks? You know, so Claire, you and Ravi were both sort of highlighting, you know, that potentially there could be risks of collecting, you know, unknown samples to study. And one question I'm interested in is what you see as the lessons that we can draw from what actually happened with the COVID-19 pandemic for research going forward. So for instance, when we look at the COVID-19 pandemic, despite a lot of interest in, you know, trying to identify potential zoonotic viruses ahead of time, COVID-19 wasn't identified until it started to spread among humans. We didn't succeed in identifying it ahead of time. So you could argue that was a failure of the scientific approach we were taking. On the other hand, I remember back to early 2020 when you heard people say things like it will be 3 years or 5 years until we have a COVID-19 vaccine, and in reality it was really only about a year until we had a vaccine rolled out that was starting to be distributed around the world. And I guess in general, one of my questions for each of you is when you sort of look back over what's happened with this pandemic, now that we've actually lived through one of these types of events that we're all trying to work to mitigate, what are the lessons you draw about how we should be thinking about what to do going forward?
Well, I think you could spend hours in discussing that. I want maybe to take two examples. One is teaching and outreach. I think we live in a world of polarization now. I want to bring one example from my university, where we have a nice course that's called the Host Pathogen Colloquium, where we pick papers and discuss with students. And I usually pick papers that are controversial, that touch the topics of biosafety, biosecurity, and things like that, gain of function work. And it's a little bit surprising to see that the students don't know that there are any regulations. So there is also, in the public opinion, people think these scientists could do what they want, and it's not like that. So I think that's something where we have to reach out and tell people how it is and what type of regulations we have. And then, of course, we have to discuss, is this sufficient or not?
Mm-hmm. Yeah, that's a good point. And I do think it kind of emphasizes that forums like this that are bringing these sorts of exchanges are, in my experience, pretty rare. Mustafa?
Yeah, I guess that my point is, we know how to do the work to describe the virus or to describe viruses that are near the jump or viruses that have spilled over already. We know how to assess the risk of each of those activities and what we need to do to reduce the risk to zero. It could be reduced to zero. What we don't know, actually, is that we are not putting enough pressure in ensuring that the training for the people and the acknowledgement for the people that are doing those experiments or those expeditions or those field trips. I mean, we know what type of PPE you need to do to go to a cave and collect samples with very low risk. But if we don't follow those, if we approach that with low level of training, that's where we are failing, is enforcing our knowledge. Because we already know we have generated biosafety laboratories at level 3 or level 4 that could handle that risk. we should be able to understand that if what we are doing is describing the virome, we don't need to collect a live sample. We can inactivate the sample at the moment, and there is zero risk because it's completely inactivated, and we are only recognizing the nucleic acid in that sample. I mean, we know how to deal with the risk. What we are lacking is in professionalism.
And so what, I mean, I know Volker was talking about this course he teaches. What would you see as the things that could, you know, what do you see, Gustavo, as the things that are needed in terms of training of the scientific community or of researchers on these issues?
So, I mean, to make some examples, we know it's not under dispute that some events in that work that should have been done at the higher level of biosafety was conducted or was being conducted at BSL-2 conditions. That is not disputed. I don't know, I want to take, so one of the comments was, well, that you made was actually that the information that was collected before the pandemic was not useful to control COVID-19. Well, that is our fault. I mean, RAG-13, even when it's different at the 96, 97% level compared with SARS-CoV-2, could have been very well used as a method to explore how to make a vaccine, and probably a vaccine that could have the spike against these viruses could have been at least grossly protective against SARS-CoV-2. We could have, I mean, we knew that clade of viruses was there and was circulating. We just did not take any action to use that information to prepare. But the information was there. Sorry.
Yeah. And Claire and Ravi, what would you say to those questions?
It goes back to the comments I was making about, you know, having an approach which does high-level surveillance, intensive surveillance, and then acts upon it in a concerted fashion. But that will take, you know, a large amount of cooperation and funds to do. But that certainly is something that I think we should be advocating for, because that will be the ultimate application of science, really, to generate an array of vaccines, even antiviral compounds, against an array of viruses. So I think that's something positive we can at least advocate for as a community.
Yeah, I think it's very hard to predict, though, what, you know, the next pandemic will be. I guess in hindsight, everyone would say, well, we should have predicted SARS-CoV-2 in a way because coronaviruses are particularly good at jumping species. They've done it before. Influenza viruses are very good. So I guess more broadly, I think there were a lot of general positives that came out of what I think I've learned, and, you know, hopefully my colleagues have learned as well, about you know, engagement, data sharing, openness, transparency. You know, I think we saw the best of science in how scientists collaborated and openly communicated in the response. So I think, you know, trying to identify what are the likely potential pathogens is worthwhile, but having processes like we do now in place to respond even while we're trying to figure out the biology. You know, I think we did a pretty good job at lockdown, engagement, responses, social distancing, all of these sort of containment measures that got, you know, the R number down, that bought us time to understand the biology of the virus, to get a vaccine and to start to, you know, have some therapeutics. So I think that kind of framework is, you know, how we respond to a respiratory pandemic, how we might respond to a pandemic of, say, Ebola-type virus or Mpox. You know, we can have different frameworks in place for how we would respond to that if it was something unexpected.
Yeah. And Ravi?
I'll just follow up. Yeah, I mean, it's interesting. We got away with it for SARS. I mean, obviously, millions. You know, we still lost a lot of people, but the vaccine, as you say, took something like a year to bring to market. but we used lockdowns, for example, and other methods to slow things down. Now, the next pandemic, you know, we're already talking about whether we should have done a lockdown, right? So the next time around, the public won't accept a lockdown, let's say. So we won't have as much time to get these vaccines, you know, together before before large numbers of people die. So I think that's also a thing to consider.
Yeah, so and certainly public health responses is a very difficult question. I guess I want to return quickly to thinking about the vaccine. So thinking back to what David Relman was talking about in his remarks at lunch before we started this, is he was talking about how now, due to these new capabilities, sort of many researchers can take the sequence of a virus and just create the actual physical instantiation of that virus using reverse genetics. And he was talking about some of the potential attendant risks there. On the flip side, I would point out with mRNA vaccine technologies, we saw both academic researchers and probably more importantly, companies like Moderna and Pfizer having that same ability to simply take the sequence of this spike protein and essentially have a template vaccine in a matter of weeks, whereas, you know, in the old days, which were not that long ago, it took a long time to produce a vaccine. And I really think it's interesting to think about this as sort of two sides of the same coin. Many of these technologies that David was talking about, the ability to do rapid gene synthesis, to identify and sequence these things, also gives us the capacity to make these vaccines in the same way it gives someone the capacity to make the actual viruses. And I guess, do any of you have viewpoints in how the thinking about those new capabilities should sort of change how we think about preparation for both zoonoses and biorisks?
Well, I mean, it is all related with transparency. I mean, the information, but the quickness that you get information that you can use depend on that information to be available to everyone or publicly available to people that actually could act in generating a vaccine. So we need a new system to encourage that transparency. We still, for COVID-19, we still do not have full transparency from all of the actors. So that obviously it's a huge problem that essentially if you keep for yourself certain information, I mean, then all the good things that could come from the action from that technology could not be usable. Now, other fields, and I like the idea of the Bulletin of Atomic Scientists to take the lead on this, is because in the nuclear aspects, we have deal with these type of problems. There is an international agency that essentially handles and supervises and look at what all the states are doing, ensuring that there is openness, ensuring that there is transparency, ensuring that when a state do not help, I mean, it's a non-cooperative state that could actually be sanctioned by the international community when it's not open and transparent. That needs to be, that should be now the norm probably to other areas, like in the area of biosecurity.
And just to sort of elaborate on that analogy that you're making there, Gustavo, I mean, I think that the Bulletin of Atomic Scientists also provides a model in terms of scientists thinking about the responsibility of this. But on the other hand, when I think about many of these capabilities, both the capability to make at least a preclinical vaccine or the ability to generate a virus, it's not like producing a nuclear weapon, right? There's multiple people just sitting up here on the stage who would be able to take the sequence of a new coronavirus or new influenza virus and both produce that virus in their lab or produce a vaccine candidate against that virus in their lab. So it's sort of a highly decentralized capability, which I think we've seen many of the ways that it can be used for benefit, but you also don't have have to have a lot of imagination to think of some of the ways that it could be used to detriment. And so how would you think about that if this is not something like nuclear technology where it really requires highly specialized, high-speed centrifuges and uranium, but it requires, advanced biotechnology, but the type of biotechnology that all of us have in our labs, as do thousands of other scientists around the world?
And maybe I can comment on that. So first of all, let's say you generate a virus from a sequence that is essentially reflecting the wild-type virus. So I don't see a difference between this copy and the wild-type virus in terms of biosafety. So the same phenotype is expected, and you treat it with the same safety measures. So what we discussing a lot is this so-called gain of function, where everybody has a little bit of a different view where it starts and where it ends. And here, anyway, the scientists are responsible to tell the authorities what they are doing, what risk they think this may have, and then it's weighted against benefits. And we have to come, and that's also the purpose of this meeting, I think, to come to some ideas how we evaluate that, where is there any natural border that we should not cross? And that's maybe a constant thing. It's maybe not even what we think today may not apply tomorrow. We may shift our view or change our views where the borders are and what we should do. And the other thing is what people often think is that you have a sequence, you have a virus, you design something, and then you have the desired phenotype. It's not like that. We don't know enough. Whenever you study those functions of a virus by reverse genetics, you're surprised how you can mess up things. So in terms of the virus doesn't grow anymore, it just is not viable. It is so creating something new is, with our knowledge, barely possible. So the people probably think we are better than we are.
Yeah, that makes a lot of sense. I agree with that.
Yeah, that's a really good point. I think any of us who work on viruses know it's much easier to break the virus and kill it so it doesn't do what you want anymore, it doesn't replicate, than it is to give it a new or enhanced function. And I think you're right that the conversation absolutely needs to be had, but gain of function is not one particular area of virology. And what is your gain of function in antibody escape, for example? You may view that as gain of function, but it's incredibly important to know how a virus might evolve to escape a current vaccine or might evolve to escape a drug. So anything that changes the biological properties is not necessarily gain of function, and that gain of function research is not necessarily a bad thing. And I think that's a really important conversation that we need to be having with regulators, funders, the public, and I think this is a great forum to start that conversation, because I think it's become a very loaded phrase without people really understanding what it means, and a one-size-fits-all regulation is not going to work in this regard, I don't think.
And just to add to that, yeah, I agree with all that. So just to add, the layer of sort of oversight at university level, of course, varies between institutions, and this is quite a big burden, of course, for a committee, as David was mentioning earlier, to have a proposal, and you're having to weigh up the pros and cons, and potentially the level of expertise or experience on those committees will vary, and some will be conservative and then say no to things, and some you can imagine being more liberal. So I think that is a problem, and so that's why I think actual, so explicit guidelines are really quite important that could be circulated.
And to sort of get to that more specific question and allude to what a number of you were saying, because I completely agree that this term, gain of function, is an extremely broad term that very many things, perhaps a small number concerning, a very large number probably not concerning, fall under that umbrella. How would each of you think when you think about your own research and how to draw these lines when you're working with different viruses? And as I ask that question, I'd even specifically call out 3 different types of viruses, broadly speaking, that you could imagine working with. One is a virus like SARS-CoV-2, which is widespread in humans. Another is a virus, like we were discussing in these virus discovery and looking for the next potential virus, which is a virus which hasn't infected humans, but who knows, maybe it could. And then the third category might be something like the 1918 or the H2N2 influenza, which is a virus that we know can infect humans, but isn't in humans right now. And I guess I'm curious how each of you would think about, in your own research, how you would kind of put these cost-benefit analyses when you think about that type of research.
So the rule that I have been following, and actually that I take from the risk assessments done at USAMRIT and for Dietrich, is that you only should work with something that is currently circulating in nature. If it is not currently circulating in nature, there is no need or reason to work with that pathogen. If we are changing something on a virus that you have not observed in nature, a change in an area, you're generating essentially a chimera that will need a different level of risk assessment that probably should not be in the hands of a scientist to decide. It should be at a higher level, or where you don't only have scientists, but you also have regulators, biosafety experts, and biosecurity experts. And I think that that is a reasonable, I thought that that was the rule everywhere. And again, I go back to that nonchalant state of assessing the risks that I mentioned at the beginning. I think that that is the major problem. If we are not aware of the risks, there is no way that you can minimize them.
Any other thoughts on this topic?
I mean, I would agree. I think making something, working on something that exists in nature, or modifying a virus in a way that reflects a natural circulating COV variant, for example, because you want to understand function of a gene. Yeah, I think mixing spikes, creating a whole new virus with altered tropism is very risky. And I think people need to, there's experiments you can do, and then you need to sometimes reflect and think, is there, something you and I were discussing yesterday, Jesse, is there a better way to do that experiment? Here's the first option to do that experiment, which is modifying the virus. But what if I modified the cell, for example? Is that a safer way to get to the same answer? And I suppose, you could think of it from the worst case scenario. If something went wrong, can I justify having done that experiment? Can I justify having made that viral mutant? So I think you need to just pause, but just because you can do the experiment doesn't always mean you should, and reflect on, if something was to happen, am I comfortable that I could defend the work that I was doing was absolutely necessary?
I mean, to add on that, you said changing, for instance, the spike in the coronavirus. Also here, the borders are not clear. For many of the variants, we're talking of a handful of bases that are different.
Oh yeah, I meant something more like putting MERS spike, completely different virus. Yeah, absolutely.
Exactly.
Yeah,
so also here we have to
that's
consider.
Where it's very grey, isn't it?
Exactly. Yeah.
And so I hear in some ways Volker and Claire coming down on this criteria, which has actually often been used in the regulations. Are you making something that could reasonably expect it to occur naturally? Is that almost what you're suggesting? It sounds like. Ravi, you want to?
Another way to of course approach this is to have reverse genetics as your last set of experiments, and you start with pseudotypes, for example. So you don't have to go straight to a live virus to demonstrate or to explore the scientific basis for genetic changes. You can just start with simpler systems, virus systems, using the spike protein on a lentivirus, for example, and just manipulating that, which is completely safe. And you can find out 80% of your facts that way, and then you can make the live virus if you can justify it.
Yeah, and I do think that Ravi makes a very good point there in the use of, for instance, in studying coronaviruses, you can make pseudoviruses which, don't pose a hazard to humans. And we've also seen, many examples where researchers have taken these same technologies we're talking about and, for instance, created even replicating coronaviruses, but where they've made intentional modifications that will attenuate those viruses, such as removing key accessory proteins. And so I do agree that perhaps in many cases there's ways that we can think intelligently about using the technology to actually make these experiments safer? I don't know if you want to elaborate on this. I know we were talking a minute ago about work that you were doing.
I mean, that's a classical example of if you want to study a function of, let's say, a viral gene, you would knock it out, for instance, and you would expect loss of function here. Like you said, a virus that has lost one function. And here I don't see any critical points.
Mm-hmm. Yeah. Yeah, so to go to this question where we can, think about sort of in what are many cases these gray zone judgments, like is this a virus that would occur naturally? Is this risky? Is there a safer way to do this experiment? I guess the last big question I have for all of you, because it's a question that I wrestle with a lot, is sort of how do we have these discussions? There's a lot of controversy around this. As you know, there's a lot of people in sort of our own field of virology that sort of feel like we're under attack as being irresponsible. There's a lot of people out there in the public that feel like virologists aren't thinking carefully and are being irresponsible. And it's become, in some ways, a very non-productive and conflict-prone discussion. And what do you think are the things that we can do and can encourage to sort of improve the quality of that dialogue and recognize, how we being both virologists and people worried about biosafety, work together to sort of get to a better place?
I mean, I think that's probably one of the most important sort of questions there is, in terms of how do we move on from here? Because clearly basic science and virology has been immensely important in human health with pandemics, throughout the last 50 years or more. So we need to continue this type of work because if it doesn't happen, of course, we'll be blamed for not doing it down the line, number one, of course. And there is, a moral imperative in a way. We have the tools to improve human health and prevent disastrous pandemics from killing people. So I think it's a process of rebuilding trust, number one. Communication, of course, but this is really hard now, of course, with social media. To get the truth out now is much harder than it used to be, probably, because there are so many voices from different directions. So again, a coordinated effort it's going to have to be a supranational organization, I think. It's going to require money, but I think investment is going to be critical because, of course, you're looking at public trust in science at the end of the day, and I think it's in government's interest to promote that. So a concerted sort of effort to rebuild trust in the scientific process is important. We can remind people about vaccines and how, and actually the benefits they've brought. Of course, there are counter arguments from people about, side effects. So there are a number of issues that need to be kind of taken one by one, and the facts kind of replayed to the public because, of course, we're now in a situation where we're hearing stories about why we shouldn't have done lockdown or why we shouldn't have closed schools. There's a lot of that revisionism going on because people have forgotten about the things we went through. So I think that that this is a really important time for the world's public to be reminded of what science did during this period. And then once you've got there, then we have to say, well, we'd like to stop this happening again. And of course everybody will buy into that. Because I don't think some people did benefit from COVID-19, but most people didn't. But I think there's a period of rebuilding trust that's essential.
Yeah, I would second that. I think somehow trust has been lost, and I think social media and misinformation is not helpful there. So I think we have a responsibility to engage and educate. And I think Ravi made a very good point about it being a collaborative effort. So I think this type of meeting is a fantastic beginning. So we have responsibility as scientists to behave responsibly. Journalists have responsibility to report responsibly. And then above that, you have institutional and governmental responsibility, but something as well that we could think about is, a responsibility or an interaction with funders and reviewers of grants and reviewers of papers. I was talking to someone the other day about how when you just kind of making sure that people are aware of there's a responsibility at every level to reflect and audit and make sure that things are done safely and promote, the positives of science. So it definitely has to be a collective effort.
Any thoughts, Gustavo or Volker?
I feel it's a difficult point for me to try to explain. I am a virologist and I am a scientist, but for the last 10 years I have been working in government in a biocontainment laboratory and handling risks and biorisks. So I have seen the other side of biosafety and biosecurity with a heavy impact on what I have been doing. One of the feelings that I have about what's happening in all of these discussions to scientists is that as scientists, we are not listening. The community is responding in a way that we are the expert, trust us. But we have not engaged with other parts of the communities during this whole process, where we accept that there are other points of view. The reconstructions of the majority of the discussions on the origins of COVID-19 demonstrate the absence of experts in biosafety and biosecurity and biodefense in all of those discussions. We were setting policies or discussing how we were going to deal with the more likelihood scenarios, but we were not involving other people. Most of the discussion was among virologists and evolutionary biologists. So that is not listening, and that's why we are in the situation that we have lost trust from the public.
Any other thoughts?
I can echo what has been said, but maybe something more towards the future. I mean, things are always seen in a certain way at the time you look at it. So now, as was mentioned, people may not consider that a lockdown should be done anymore. And at the time we had the lockdown, people thought, oh, I want to be safe, I stay home. So we have to be clear that now we are coming out of a pandemic, and we see things maybe different now than we will see it in a few years down the road. So this process that we are starting here, or that we are having here, we have to revisit that. So we have to think about where are our borders, how should our regulations be, but we should not be too impressed now from the pandemic. Next pandemic might be something completely different. We may not get a vaccine because a simple vaccine like mRNA is not working. You know, there are certain things that we have to think about beyond this current pandemic, and have to consider that things are maybe different in a few years from now.
Yeah, I think that's a very good point. I don't know if anybody's ever read the report on the 1976 swine flu scare, which was a pandemic that never happened. But one of the points in that report that's always stuck with me is one of the people wrote, pandemics only happen every few decades, which mean that scientists get to make very few observations and have a lot of time to think about each observation, and that's a circumstance ripe for overinterpretation.
But we should think about it and not forget, because people also forget in a few years from now. I mean, you will see that funding is going down. People are not interested anymore. So we should keep that up and discuss.
Yeah, so I agree. And to sort of, I think these are all good points, and to sort of move on to Gustavo's point that part of our job is to try to explain our perspectives, and part of our job is to also try to listen to other perspectives. For the last 15 minutes or so, we wanted to kind of open the floor for questions, if there are questions. I guess I'll just call on people if there's not another system. Natasha, go ahead.
I'm taking my question. Could you tell me what you think the risks and benefits of the global Virion project are, and whether it's worthwhile to pursue it?
Okay, so Natasha's question was specifically, and it relates to some of the topics that were being discussed here, the Global Virome Project, which is a large-scale project to sort of catalog the diversity of viruses that are out there in nature, perhaps with the eye towards identifying the next virus that might cause a pandemic. Does anybody on the panel have thoughts on this?
Well, I mean, it's an essential project, right? We have the technology to do it, and many of those viruses may have human benefit. So it's critical that we do understand what's out there, but in a safe way. So as we mentioned, if you're just going to look for sequence, you can inactivate the samples and just have nucleic acid, and there's no infection risk. So if that's the aim of the project, then great, because you could always then resynthesize or look at other ways of examining those viruses that you're interested in. At the moment, you're going to try and catalog 10 billion new viruses or whatever, and that's purely a sequencing exercise. So for me, there's no risk from the Virome project.
Maybe also commenting on that. I mean, we have maybe more risk in things we already know, and this was mentioned before. We knew that a SARS-like virus can emerge in people. We knew where it comes from. We knew from projects that sampled in bats that there is a high diversity of those viruses out there. So we knew all that, and I think it's to mitigate risk is better to then act and say, okay, we know that they are all there, so we want to prevent that they are transmitted from animal to human.
What's that? Oh, take a bunch of questions? Okay.
You know, the word trust has been mentioned several times, and it has many dimensions to it. One of it is the understanding of the scientific process. You know, scientists sitting in their ivory towers never really explain how the scientific process works. It works on the basis of data and how, you know, data has to be transparent, and that data to transparency builds trust. So, you know, the fact that WHO says masks work and then say masks don't work, the public has this perception that when a scientist says something, they're speaking the truth. But that truth also depends on the technology available. That truth also depends on many other circumstances. I think understanding the scientific process is so important, and this is something that each one of us, scientists or not, has a responsibility towards.
Yeah, I think you're absolutely right. Trust and education go hand in hand, don't they?
Hi. Following the discussion about trust of the population, we have talked a lot about transparency from researchers, from academia and governments. However, should we also ask transparency from the big companies? And sometimes, when can we draw the line between knowledge that have to be shared having a public health implication? What do I mean is that in this pandemic, we have seen that quickly academics have shared sequencings that then were allowed to do PCRs and have a rapid response. However, this same knowledge helped to have quick vaccines. But once we have the vaccines that work well, they're all on profit problems. And this is an issue that maybe is also we need to discuss, because it goes trust down, especially of inequality, of access, of all the response during the pandemics, seeing access for diagnosis as well as access of vaccines at treatment.
Hi, Tim Trevan at the back here with Coronavirus Risk Management. Not questions, but two quick comments. First, on regulation. Part of the problem with biology in general, not just virology, but biology in general, is the field is moving so fast that I don't think regulation is the solution here. It may be a component of the solution, but it cannot be the solution, simply because the regulators don't understand the subject to regulate it before it happens. So, second of all, if we regulate it, and in Germany may well have very good regulations, but other countries don't. If you regulate in one part of the world, water flows where it flows. The science will go to where it's possible rather than where it's regulated. So regulation and engineering also doesn't really work. Some of the biggest incidents we've had in the field of biosafety and biosecurity risk have been in the most competent, most funded, most engineeringly advanced facilities. We have to take into account human behavior. And that means that we have to start thinking in terms of responsibility at all levels of the organization, as Claire says, and we have to look beyond biology to how other organizations in complex adaptive environments manage their risks. We have to look at high reliability organizations and what they do in terms of creating the ability of everyone in the organization to speak up and to challenge anything which approaches an issue which might have high consequence. We have to organize where consequences are high to prevent those risks from happening.
So I'd like to follow up a little on that point, because I think this is a good point. I mean, I think even in our discussion here, it was clear that none of us who are supposed experts have written down exactly the solution to how this should be regulated. And I think the point that this is a really fast-moving field, which makes it even more challenging, is a really good point. So I don't know if anybody has any comments here. Like, what do you think we can do to create a dynamic environment which sort of promotes exchange and dialogue on these topics when it's very hard to write down a static set of rules which is going to stay current?
It's a difficult question. We heard about trust. We heard about things that are probably outside our academic world in industry, and regulations that are different in different countries. I think you could probably provide some guidelines as sort of a minimum, some advice. But we know from many fields, not only from biology or medicine, that regulating something globally is close to impossible.
Yeah, and I do think, to go to your question, the analogy that Volker just made with medicine maybe has some merit. I mean, the field of medicine is advancing very rapidly. There are regulations that grow up around it, but they're changing all the time. And I think figuring out how to apply that same sort of flexible thinking, recognizing risks and benefits, is an important question. Are there other questions in the field out there, sort of?
Very quickly, because I know we're nearly out of time. I want to take issue with something I think you all agreed on at the beginning. Volker said that there is much more contact with wildlife these days. I don't think that's true. The rural population of the world is falling now as an absolute number as well as a percentage of the population. And even in rural areas, contact with bushmeat, wild meat, as opposed to farmed meat, is rapidly falling. So the vast majority of people are not getting their food from wildlife anymore or other contacts. And in terms of encroachment on wild habitats, again, the world is reforesting now, not in many places, but in a lot of other places it is, and in particularly southern China, there's been a rapid reforestation. So the amount, people are moving out of those rural areas into cities where they're having less contact with wildlife. And this was brought home to me when I had a conversation with an archaeologist who said, compared with our Neolithic ancestors who were obsessed with going into caves, we have way less contact with bats these days than we did then. By contrast, of course, lab risks are increasing because of the number of experiments and the amount of work going on. Just want to introduce a note of disagreement in that.
I think in this science of viral spillovers, essentially, you have two steps. The first one is the contact with the reservoir. The second is secondary transmission, human-to-human transmission. So even when you are reducing the first step, because you are right, that there is maybe less contact, the increase in human density in all of those places increases the chances of a successful spillover as part of that two-step process.
Yeah, I would agree with some of what Gustavo said there. And I recognize Matt's point. I mean, from my perspective, an important aspect is, regardless of exactly which risks are going up and down, they're all real risks, right? They're all substantial risks, both wildlife, farmed animals, and biosafety incidents. They're all real risks. And, to some perspective, arguing which is a more real risk than another may miss the point that they're all real risks, right?
We didn't really. I don't know if we answered the question about pharma and organisations and their their level of whether they have to report incidents, etc. That is quite an important thing because, of course, pharma are working in infectious disease areas very actively, and they're testing compounds. They're testing for evolution of resistant strains, and so we could regulate science in universities, let's say, but how does that feed through to organizations? And that's quite a big challenge, but it could be an opportunity, I think, of course, because university pharma partnerships are a big thing now. So, but I think it still needs to be on the agenda as part of any strategy. How do we capture work going in different areas?
Yeah. Go ahead. I don't know who has the microphone, but. I think there's a question over there.
My name is Jonathan Latham. My question is for Volker. Where do you draw the line between countries that obey the law in your terminology and countries that don't?
I cannot draw that line. I mean, that's exactly our problem. I can just tell what in my environment, so I'm working in Bern in Switzerland, so I have the feeling here everything is very well regulated, and I think that's probably in other countries, or not all other countries, it's regulated to the same extent. So for instance, we don't have institutional biosafety committees. We have it on a national level. So every application goes through the same channel. So these kind of things. But where you draw the line is difficult, because then you also go into politics, and it's hard to tell any country you're doing bad, and they will say, no, you're doing good. So it's very difficult to draw the line.
I mean, just one approach in terms of the building trust and kind of managing risk could be, for example, I mean, you have national clinical trials registries, where if you're doing a trial, it's available on the web and you can read about it. You could envisage a situation where, to build trust, we submit a proposal online so that everyone can see the experiments you're doing, and the mitigations you have in place and the risk management plan, for example.
Yeah, I do think that's a good point. I wanted to quickly, since we're wrapping up, we just have a few more minutes. I wanted to go through the panel and sort of if anybody had any last comments they wanted to make. I know you were about to say something, Claire.
No, I was just going to say, I think it does differ between countries, and certainly, in the UK we have university level regulation and then national level regulation. But I know that what is containment level or BSL-3 in the UK is not necessarily BSL-3 in the US, for example. So I think we all agree that there are risks, but even amongst ourselves and the experts and the regulatory bodies, there are differences in assessing risk. So a good starting point might be to sort of unify that framework.
Well, a bigger challenge for me is the fact that depending who's funding your research, you need to follow certain regulations or not, which that opens a lot of big kind of worms.
Yeah, but that's what I mean. That's certainly different in different countries, because in some countries, including Switzerland, you don't need so the regulation or how you should conduct the work under which biosafety conditions is not dependent at all on funding.
Mm-hmm. I mean, something that Ravi mentioned, something that we've talked about before, is that, if you want to do animal work that is regulated, you have to justify in a grant application your numbers, why. Me as a reviewer, I would have to then comment on whether I think it's justified. You have to do the same thing with human work in clinical trials. There's no box on the form for a reviewer of a paper, in many cases, to comment on the virology research. There's no box in many grant funders where you have to justify the experiments and the reviewers comment specifically. So just that sort of level of at least getting people being forced to think about what they're doing, and people who are reviewing it think about, might be a good first step.
Yeah, and this is a very interesting point that Claire makes, and I think it relates a little bit to what Ravi was referring to earlier in terms of sometimes using pseudoviruses. For those of you who aren't involved in research, you may know if you're doing both animal research and human subjects research, you're supposed to justify why you need to use those animals, or why you need to do these experiments involving humans, and why there isn't an alternative way to do it. And obviously in many, many cases, we do use animals or do experiments involving humans because there isn't an alternative. But for biosafety, there's currently no criteria that you justify why you're doing these experiments. And I do think this point that perhaps it could be thought about in that way is one at least worth considering.
I agree, and it's fairly straightforward. It's something you could action immediately. And we know that the funders do get together, and for example, for open access publishing, Wellcome Trust came together with HHMI and various, a few others, and maybe NIH to make that happen. So there's no reason why they couldn't do the same thing for biosecurity in grants applications, especially with the amount of scrutiny that funders came under during COVID-19 for the work that was going on in Wuhan. So
Yeah.
I think that there is an impetus, and maybe they're already sort of planning it.
Anyway, I think we have about 22 seconds left according to the clock I'm looking at. So I'd like to thank all of you for coming to the panel. Before this began, I was often asked, like, how are we going to resolve all these questions? And I don't think we resolved them all, but I do really believe that, having scientists coming to these forums and really discussing these issues with people from diverse perspectives is an important step. So I'd really like to thank all of you for that. And we're now going to transition to the next panel, which is Global Biorisk Management.
A very warm welcome to the second panel, and a very warm welcome from me to the conference, to all of you in the room and to all of those of you joining us online. My name is Philippa Lensos. I'm a social scientist based at King's College London. I'm also co-director along with Jessie Bloom for this Pathogens project. It's very exciting to finally see you all here in the room. It's been a long time in the planning, and I'm glad we've got off to such a great start. With me on the panel today, we have Sam Bonti Ankoma at the end. We have Alex Demarche. We have Adeji Ebo. We have Stephanie Norlock and Emmanuel Turlings. Our panel will be focused on global biorisk management. The panel just before us already got us off to a great start in thinking about biological risks, of which there are many and many different kinds. The previous panel talked about biorisks in the future primarily. This panel will talk about biorisks in the present. And the way we think about biological risks generally is risks related to safety. So how do we operate safely in laboratories? How do we make sure we don't infect ourselves and others in our community? It also relates to security. How do we make sure we secure the pathogens that we're working with? And it also relates to dual use. Dual use is one of these incredibly difficult terms to wrap heads around. I think of this generally as relating to the possibility of repurposing the science for alternate uses, potentially for nefarious uses. Another way in which to think of dual use is in terms of capacity building and competition when it comes to capacity building and building capacities in countries in terms of, for example, infrastructure, lab infrastructure, scientific expertise, and the possibilities that this enables for misuse. So that's a little bit about how we think about biological risks. Risks cannot really be eliminated. We can't bring these risks down to zero, and this is why we talk about management. So the focus of our panel is bio-risk management. How do we deal with this variety, this plurality of risks as a global community? And current efforts at dealing with this are, there are current efforts, that is the one good aspect of this. There are lots of current efforts at dealing with biological risks. And there are institutions, global institutions, that deal with this. There are informal ones and there are formal ones. Some of those institutions are present on the panel with us today. We've got the WHO present. We have the UN Office of Disarmament Affairs present. We have the International Experts Group for Biorisks and Biosafety Regulators present. We have the International Federation of Biosafety Associations. We have private industry present on this panel, and there are many other institutions that also try to tackle biological risks with us in the audience. So there is this whole range of views that are present. So over the next 70 minutes or so, 68 as the clock is nudging me on the bottom here, 68 minutes. Our panelists will elaborate a little bit on how their institutions are currently addressing biological risks. We will then turn to you and collect comments and questions from the audience, just like we did in the first panel. We will also try to feed in from the online audience, so please do use the hashtag Pathogens Project, and we will try to get comments included from Twitter as well. And then finally, I'll turn back for concluding remarks from our panelists. All our panelists have had incredibly interesting career paths and have extensive achievements, but I'm going to save time on formal introductions and refer you to our conference website where you can read more in depth in your own time about their backgrounds. So without further ado from my side, let's start. And Emmanuel, I thought I would start with you, Emmanuel, from the WHO. In 2 minutes or so, what tools and mechanisms does the WHO currently have to deal with or to address biological risks?
Thank you. So WHO works worldwide to promote health, keep the world safe, and serve the vulnerable. And today I would like to focus on the work done under the stewardship of the WHO Science Division in close collaboration with our WHO Health Emergencies colleagues and Global Health Ethics on the development of the global guidance framework for the responsible use of the life science, mitigating biorisk, and governing dual-use research. This document was published last year in September 2022. And I would like to make 3 points. First, on the document itself. So life science and associated technologies, they can bring huge benefits to improve our health, our societies, and our environment. But at the same time, there is a recognition that they could bring some risk, including safety and security risk. And so the objective of this document is to assist member states and key stakeholders and relevant also policymakers to actually mitigate those biorisk and govern dual-use research. This document has been developed over a 2-year process with many multi-stakeholders, over 100, including scientists, research institutions, publishers, editors, funding bodies, and others, key relevant actors, including UN entities. And some of them are here today and would like to thank them again. This document also builds upon past guidance, past experience, and also existing efforts to govern dual-use research and mitigating biorisk. And so this includes, for instance, in terms of WHO guidance, previous also guidance on global health security, laboratory biosafety and biosecurity, but also human genome editing and artificial intelligence. So the document is the first global guidance framework in that respect, also building on previous initiatives to inform national frameworks. And it has a unique kind of aspect in that it provides a set of 9 values and principles and their associated commitment to guide decision making. It provides also a set of examples of tools and mechanisms for different stakeholders, and as well practical ways to operationalize those value principles and tools and mechanisms. So the second point also I would like to make, it's 2 points that were also here in the previous session. It's 2 strategic ways to approach this. And one is saying biorisk management as an overarching framework, including laboratory biosafety, laboratory biosecurity, and the oversight of dual-use research. And the other point is undertaking biorisk management within a One Health approach. And this is in order to optimize human health, animal health, and the environment. And the final point I'd like to make is how to operationalize this now. And biorisk management is a global issue impacting all countries. And one way for us to move forward is to continue to collaborate. It's also to leverage existing frameworks, existing policies, and to create or find synergies also with other activities in the field. Thank you.
Thank you so much, Emmanuel. I think what the Science Division started developing with this set of guidelines was an incredibly important document that came out, and if you've not had a chance to look at it, please do look it up and take note of it in terms of the range of stakeholders it addresses and the sorts of framework it provides. I think certainly our project here drew a lot of inspiration from that document and also the process which you gathered together in order to develop this process. It was this reiterative coming together of groups of experts over time, and you're sustaining this now by sort of socializing the document. So I think that's one of the underlying messages that needs to come out. This is not a one-off conversation, right? This needs sustained conversation over time, and I hope this venue is one place where we continue that conversation, so thank you. Ade, I wondered if I could ask you to tell us a little bit about the tools and mechanisms that the UN Office of Disarmament Affairs has for addressing biological risks.
All right, thank you. And thanks for getting us here to the organizers, and nice to meet everyone. It's a good thing you didn't introduce us in terms of our background, because you'll have exposed me as being a non-scientist. So ever since I was in school, I've been a bit intimidated by science, yet here we are. In terms of United Nations Office for Disarmament Affairs, our role basically is that of a convener, and in terms of norm setting, we have, I believe, the architecture, if you like, for biorisk management in the UN is resting on maybe a tripod of 3 key instruments. The first one is the 1972 Biological Weapons Convention that many may know of. Now, the thing about the BWC is, I think it's fair to call it perhaps the holy grail of human conscience, because it states that the use of biological weapons is repugnant to the conscience of mankind. It prohibits the development, the production, acquisition, the transfer, the stockpiling, and the use of biological and toxin weapons. There are, I believe, 185 signatories. I'm not aware of many treaties that have such a truly universal membership. It is supported by what we call an implementation support unit. They are the real experts and the real, actually my ventriloquists. They are somewhere there in the audience, Daniel and Alex. They do very good work in terms of, supporting the BWC. So in terms of this tripod of architecture I was talking about, the BWC is the main pillar, if you like. It is also the foundation of many other instruments, many regulations, laws, and standards that I'm sure you've heard of. And its emphasis, really, and the principle it underscores, and I think we should continue to underscore, is that the use of disease as a weapon should be completely out of the question as a principle. Now, in terms of the BWC's more recent history, we had the 9th review conference of the BWC. I think this was in December last year. It was a relative success, particularly in an era in which, when it comes to multilateral discussions nowadays, countries are often not able to agree on anything. But in terms of the WC, and I think perhaps thanks to the wake-up call of COVID-19, we're able to have some success. They were able to agree on the establishment of a new substantive area of work for 2003 to 2026. That program of work would expect them to meet every year, 15 days every year. And I think one of the key achievements also of the RevCon is the establishment of a working group on the strengthening of the working of the convention. They agreed on 7 areas that they should be working on for the next 3 years, but I'm not going to go into those 7 areas to save time. Now, the second instrument, if you like, in this tripod is what we call the United Nations Security Council Resolution 1540 of 2004. And here it's about actually focusing on the possible use of weapons of mass destruction, including biological, by non-state actors, particularly terrorist groups. And what 1540 does is to ask all states to refrain from providing any form of support to non-state actors to develop, to acquire, to manufacture, to possess, to transport and transfer, or use nuclear, biological, or chemical weapons for terrorist purposes. Its mandate was extended for another 10 years, actually fairly recently, in November last year, for another 10 years. And it adopted its 20th program of work for the next year up till, I believe, January next year. And there are 4 pillars to its work. The first one is national implementation. The second is assistance. The third is cooperation with international, regional, sub-regional organizations. And the fourth is transparency and outreach. Now, we as ODA, we organize activities in support of that resolution, including through regionally coordinated approaches to implementation of the resolution. You would imagine that there are regional variations in terms of both context and capacities. So we try to create, if you like, a level playing field in the capacities of those regional presences that we have. Also, they work through, they do a lot of peer reviews and awareness creation. Now, the third in this tripod is the United Nations Secretary General's mechanism for investigation of alleged use of chemical or biological weapons, the SGM as we call it. Now, all members can report alleged use to the Secretary General's mechanism, and the SG has the authority to launch an investigation into that allegation. It is not a standing mechanism. Actually, it is composed of a roster of experts and laboratories. Right now, I think there are over 500 experts on the roster and over 80 laboratories. You would imagine that, given that this is universal and global, there is perhaps uneven capacity in terms of what experts and the labs can do. So in terms of our role as the custodian of the roster, part of what we do is to organize activities beyond outreach, but also training of nominated experts to make sure that we have a minimal level of capacity among the experts. So we have exercises for these laboratories. For example, there's a SPEES lab workshop that's going to happen in June, I believe, here in Switzerland. And there's a Ref Bio exercise workshop due for May as well. There is, in support of the SG's mechanism, what is called a group of friends, member states, which include Canada, UK, and I believe Switzerland. So I'll leave it there. I think my 2 minutes is done. Thank you.
Thank you so much, Ade, and you're very welcome to be here. You are important stakeholders. It's not just scientists. We're also very important to have all the stakeholders present, including your two ventriloquists in the audience. We've now heard from two of the very formal organizations that are part of the international biorisk management landscape, the WHO and the UN Office of Disarmament Affairs. And Ade, I think you very clearly laid out this tripod, as you called it, that you deal with, the BWC working related to biological weapons and states, 1540 focused on non-state actors and terrorists, and the UNSGM, this mechanism that the UN Secretary General can draw on if there ever is a biological weapons attack and it needs investigation. Both of your organizations, as you underlined, have this role as conveners and standard setters, and it's important that or norm setters, and it's important that these are living standards, I think, continually updated, which is why the work that you do is so important. We're now going to turn to some of the more informal institutions that are part of the kind of the global biorisk management landscape. And Stephanie, I'd like to turn to you because this is where we get a little bit more operational. And if you could tell us a little bit about the work of IFBA and the kind of very concrete things that you're doing to address biological risks in the field.
Absolutely. Thank you, Dr. Lensos. So for the colleagues that preceded me, their points seem to come in sets of threes, and I'll be no different. I have 3 solid points to present in that regard. And the first is a little bit of a description of what the International Federation of Biosafety Association is as an organization. And we are a not-for-profit, non-governmental organization that is composed of almost 50 member national and regional biosafety associations around the world. And as Dr. Lensos mentioned, it's a little bit more oriented towards practitioners. However, I must emphasize that our member associations are not just composed of biosafety officers, but they're also composed of policymakers, bioarchitects, researchers, so on and so forth. And so our national and regional associations are very relevant to the varying regional biosecurity landscapes that are relevant to dual-use concerns around the world. I'll also mention that in addition to our member associations, we also have multiple like-minded observer organizations that help us achieve our mission, and those organizations do include the WHO and the BWC ISU. And so in terms of a first point in how the IFBA addresses global biorisk, It's a little bit inherent to our structure in the sense that our members around the world have worked together in terms of knowledge sharing and translation to suit their local contexts through their communication and activities. And this is really important when we consider evolving risks, especially when you consider policies being translated off of paper into practice over the long term, making sure that those practices are suited to the capacity and also of the norms and priorities of different regions. A really good example of how those regions worked together and knowledge shared was during the early days of the COVID-19 pandemic, when a lot of laboratories around the world were lacking a little bit of direction and information with regards to how to treat samples for either diagnostics or research purposes. Our global network sort of fell together online, at first through informal networks, on IFBA Facebook pages, and then it evolved into an online portal with help from Global Affairs Canada, or rather generous support from them. And through that, they were able to kind of get practitioner to practitioner support for where there was little evidence that existed. And I think that was a really big success. An additional example, I would say, would be through the, again, knowledge sharing, where different associations are responsible for specialized professional development. So developing training for our professional certifications in biorisk management, as well as biosecurity, and most recently and relevant to dual use research of concern is cyber biosecurity and biological risk assessment. And so while us at IFBA do not perform trainings, our member associations do, and often lend these tools and resources to other associations, which really helps kind of build not just international networks of biosafety champions that can disseminate knowledge locally, which includes multiple professional sectors in government, research labs, industry, so on and so forth. But they can also, I suppose, evolve best practices and communicate with each other, which I think is really great. My second point revolves around professional development, like I was just talking about with our new certifications. We are very focused on not just focusing on the bread and butter checklist biorisk management, but we're very much embracing these emerging biorisks and looking to biorisks of the future, especially as one of our introductory keynotes was mentioning, it's not just about handling biological materials anymore, but it's also about information hazards and dual use of technology. And a final point, which is a little bit closer to my own portfolio, is about ensuring that underrepresented professional communities in biosafety and biosecurity professional fields have equitable access to information and equitable access to meaningful participation in work. And what I mean by this is essentially populations such as women and youth who may be underrepresented in local disarmament fora or decision-making capacities, they perceive risk differently, they have different priorities, and they have a lot of ideas that they want to bring to the table. And this has been reflected in limiting emerging research, a good example being attitudes towards antimicrobial use and alternatives. And it's necessary that we allow them to essentially have this meaningful participation to ensure that the collective perception of risk and responsibility for mitigating it is ensured. And I'll leave my points with a continuance of the analogy that was used in our introductory keynote during lunch of the lamppost. Driving down the highway with all of the lampposts, I kind of see our international network, or us as a collective, as all of those little lights that light up along the runway, guiding a plane to take off. And alone, they blink, and that's great, but it's that synchronous multitude of lights that really gets us to take off and to progress as a society. But we need to make sure that all of those little lights are synchronous and that they're included and that they all have a power source and they need what they need to work. So in any case, I suppose I could summarize all of my points as IFBA is very invested in producing or in facilitating the cultivation of a competent, sustainable workforce in biosafety and biosecurity that is suitable to their local contexts. Thank you.
Stephanie, I absolutely love this quote of synchronous, or this metaphor of synchronous multitude of lights down the runway. That's absolutely what we need to be aiming for, and the service that your organization is providing in terms of equitable access to fora, in terms of providing this professional development, certification, all of this so incredibly important. We heard in the first panel about the importance of education and awareness raising and training. These are the guys that are doing that globally. So an incredibly important player in terms of global biorisk management. There's another player that's still building, but we're hoping you will expand, Sam. This is not the trainers, not the educators or the professional certifiers. These are the regulators. Because how do we connect the regulators in different countries? Well, there is a network for that too, a little runway of lights connecting regulators in different countries. And this is with the wonderful acronym that just rolls off the tongue of the IEGBBR, which I'm hoping you will tell us a little bit about, Sam.
Okay, thank you very much. I think, hello? Okay.
You're good.
So thank you very much. I know it's difficult to pronounce IEGBBR. I have to slow down a little bit to do that. And so basically, yes, the International Expert Group of Biosafety and Biosecurity Regulators was set up around 2007. Basically, there are 11 regulators that are involved in this from 11 countries. And these countries all have very established biosafety and biosecurity regulatory oversights. And so they come together on a regular basis to discuss, and thanks to Global Affairs Canada, funding has been made available to help this particular group grow. And basically, to just give you a bit of background, if you need more information about the countries, you can go to iegbr.org, where you can get all the information about the organization. So basically, the main objective of the organization is to basically strengthen global biosafety and biosecurity oversight systems. And we do this by sharing knowledge and experience among all those countries on biosafety and biosecurity related to human and animal zoonotic pathogens. So that is one of the first activities we do here. We also try to strengthen collaboration and convergence of biosafety and biosecurity regulations across these countries. And then finally, we do provide capacity building materials that can be made available to any other country that would like to actually develop or strengthen their oversight systems. Okay? So basically, it is a very good organization that will be available to help other countries develop and actually strengthen their oversight systems. Okay? At this particular moment, we do have certain things that have been done already that have been posted on the website. If anybody is interested, you go to iegbr.org, you can get all that information. First of all, when the COVID hit, we tried to get together to establish some kind of information gathering to talk about how individual countries went about trying to address the challenges related to biosafety and biosecurity as a result of COVID-19. So there's a document that has been put together to actually document all the activities and challenges that individual countries face and how they address that. So again, that has been published, available as well. And secondly, we also did a compendium of biosafety and biosecurity regulations, oversight systems across all these 11 countries. And again, we also recently developed the a dual use and life science program, which basically brings together all the activities that these individual countries are undertaking to deal with dual use and life sciences. Okay? So again, these are all being put in what we call a mobile app. So again, if you have a phone right now, you can go and see IGBL app, and that will come up where you can get every information about the dual use of all these countries, a dual use oversight system for all these countries, and also all the biosafety and biosecurity oversight systems that we have in all these other countries as well. So that information is available in the mobile app, people can have access to. Right now, thanks to Global Affairs Canada, we are trying to expand our scope to ensure that we can provide more capacity building to developing countries. And so we started talking to particularly Africa CDC to talk about what kind of activities they would like us to introduce to help them in terms of working with their oversight systems or strengthening the oversight systems. And the whole idea here is to help countries be able to meet their BTWC obligations as well as all the other obligations under the UN to make sure that we can strengthen biosafety and biosecurity across the world. So basically, that's a nutshell of what we do, and I think I'll end it here, and maybe we'll be able to answer any questions that may come up later.
Thank you so much, Sam. I think your network is a great example of the power of like-minded countries and what they can do when they come together, and hopefully this will blossom into a larger, more inclusive group of regulators, because harmonizing the oversight frameworks that we have in our different national countries is important when it comes to something like biorisk management. So thank you for that introduction. Alex, we're coming to you last but by no means least, because in global discussions around biorisk management, one of the missing pieces or missing institutions so often is the private sector. And Alex has been part of the private sector for a long time, got a wonderful example of some of the real good that the private sector can do in this field. And I'm so delighted you're here to tell us a little bit about Blue Dot and the sort of work that you are doing, the tools and mechanisms that you're drawing on to address biorisk. So over to you, Alex.
Sure, thank you. I won't speak for the whole private sector, but maybe the limited subset of social benefit small companies that I do think have a role to play in this space. So Blue Dot is a social benefit company started in the wake of SARS-1. Founder Cam Khan was severely affected in his local hospital by original SARS and made the connection along with lots of other people. We need to do a better job of monitoring distal outbreaks, understanding the risk of import to affect local hospitals, local infrastructure. So built up very impressive technology that's able to scan the internet 24/7, detect outbreaks as they occur in foreign languages. I think we're up to 400 or so languages now, thanks to some excellent machine translation tools. And then critically wean down all the noise, find the subset of potentially critical epidemics that have global implications, and rapidly forecast and assess which ones are likely to become global problems. To Blue Dot's credit, we were among, I think, the first 2 or 3 systems using similar methods outside of China to identify the early signs of COVID in late December 2019 through some secondary discussion in Taiwanese business journals of Li Wenliang's WeChat posts. So that's one, I think, great example of the continued usefulness of event-based surveillance in the modern era, particularly in environments that don't have as direct connections to the global public health community, either for resource reasons or other information sharing reasons. So I think our role compared to others on the panel is more limited in the sense of being enablers of the excellent work of analysts, investigators, other professionals, having the ability to rapidly iterate, explore new tools, having a maniacal focus on producing useful, time-saving technologies that folks can use to preserve analyst time for the really hard-thinking, investigative parts of their jobs. So we see our role as supporting all your work and are delighted to continue to do so. I mean, in the world of event-based surveillance, there are really interesting stories emerge. And I think one recent story is a useful example of the role that these kind of technologies can play. The Netherlands recently implemented wastewater surveillance in a polio essential facility, and I'm sure many of you heard of this, identified wild-type polio in wastewater spillover. This is kind of a shocking event in some ways, because this is one of the few venues in which wastewater surveillance is implemented in a very high security, high trust, excellent biosafety setting. And it was shed and a potentially very hazardous event. I think there was a lab technician who was shedding virus who resided in a relatively low vaccine coverage neighborhood, was detected by one of the, my understanding, few systems that have been implemented that do this. And I think that raises, in my view, the chance that similar events are probably happening undetected in other parts of the world. And I think that's where internet-based, event-based surveillance still has a role to play in detecting these events and potentially connecting them to facilities in release scenarios. That's something we haven't fully implemented, but are really interesting in discussing and exploring. So back to you.
Thank you so much, Alex. So, and thank you to all of you for telling us a little bit about the sort of work that your organizations are doing in the present to address biorisk management. I'd like now to turn very briefly, before we turn to you and ask for your comments, but to think a little bit about how, or to tell us a little bit about how your organization is thinking about biorisks in the future and what role your organization sees for itself in terms of keeping us safe, secure, and the research that's being done responsible. So I think we'll start again with you, Emmanuel. How is thinking about the future and the sort of risks that might be on the horizon, the sort of things that we were talking about in the first panel, being addressed by the WHO?
Thank you. So WHO strives, in fact, to keep abreast of latest development in relevant area, whether it's in research, science, technologies, in order to anticipate, to proactively identify, and then prepare for issues that hold for great potential for global health. And I'd like to focus on one tool, foresight. In 2020, the WHO Science Division established a global health foresight function with the aim to assist member states to build future thinking and horizon scanning, for instance, into their strategic health planning frameworks. And so the idea here is not only to be reactive, but also to be proactive and trying to identify those emerging technologies, those emerging issues. And so providing tools to member states in order to respond in timely manner. And so I'd like to maybe to make 2 points here, is that anticipating risk is part of good governance for the responsible use of the life science. And so as the new technologies emerge, as there are advances, the landscape also of risk will evolve. And so it's important to have governance systems that are flexible, that are enduring, and foresight applied to biorisk, to the governance of biorisk, is one tool that could be used to have those governance framework more proactive, more flexible, more enduring. So what does that mean in practice? I can give you just a few examples. Foresight is a guidance on how to use foresight that has been published recently on our webpage. There is also a training on foresight that is available on Open WHO. And in this module, in this training, there is a module applying foresight to the governance of dual-use research and biorisk. A second example is in the framework that was introduced earlier. There are also 7 scenarios. And these 7 scenarios are aimed to assist different stakeholders to also think about action in the present that would help them to face different plausible future. And the last point, the last example, is also in 2021, there has been the publication of an international horizon scan on dual-use research in the life science. And this publication is also available on our webpage. Thank you.
Thanks, Emmanuel. Actually, that resonated, I think, very well with what something that Claire was saying in the opening panel about how it should be a standard for every scientist to be able to justify why they would do a risky research. And here you are setting a standard for an organization to say that part of good governance, part of responsible science is anticipating future risks. So I think this is nice to see some of these resonances. Ade, I wonder if we could ask you how the UN is considering future biological risks that are coming on the horizon.
Thank you. In terms of the UN, the first thing we want to do is to make sure that we preserve what we have, and by which I mean that we are able to protect the existing instruments. The research is evolving, it's not static, and we also have to move along with it. So that's the first priority, to make sure that the instruments that we have are fit for purpose. The second is to ensure as much as possible a more holistic approach to biorisk management. Now, you may have heard about the new agenda for peace that is being pushed by the organization, which actually is a response to the current threat that we have, but also the emerging threat and the new domains, including biological. And here, there's a proposal to establish what is called an emergency platform. Now, the emergency platform is not going to be some new body or organization. I mean, I know that people have no more patience for new institutions within the UN framework, so we are not going to create one. What will be created, however, is a set of protocols within the UN that can be activated almost automatically when needed. Remember when COVID happened, we didn't have something like that within the UN. So this is to bring together various actors, member states, the UN bureaucracy itself, key groupings, international financial institutions, certainly civil society, regional organizations, the private sector, subject matter experts such as those here, and so on. So that within the framework of the UN, the SGI and its team can pull all these people and resources, organizations together to have a truly multi-stakeholder approach to such an emergency situation. And that is very big for the SG. And we are still working on that. I think the big message here, from what I see, even going beyond biological risk management, is this issue of this message that the UN cannot do it alone. No actor can do it alone. We need a multi-stakeholder approach. We need collaboration. We need partnerships. We need certainly synchronization, synergy. So those lights that were suggested need to be aligned and synchronized. And the UN's role, I think, as the most global of all those lights, is very important. I should also add that one thing that my ventriloquists have emphasized in our conversations is the need to go beyond the hard laws towards more soft laws. We have the BWC Convention, we have the 1540, I mentioned that, and so on. But there are also the soft law elements. I was informed, I did not know, but I was informed about the Tianjin biosecurity guidelines, for example. I think this brought together US and Chinese scientists and the notion of a code of conduct for life science. I think That's a good example of soft law. Also, my colleague from WHO talked about the global governance framework for responsible use of life scientists. That's another good example of soft law. So what is important to emphasize here is that the development of these soft laws should be within the framework of and in synergy with the existing hard laws. Within the UN itself, however, I should add, we are also thinking on about how we ourselves should be better organized and more coordinated. So responding to the wake-up call of COVID-19, if you like, something called the UN Biorisk Working Group was established in August 2020, and the UN Biorisk Working Group is actually co-chaired by the UN Office for Disarmament Affairs and WHO. And the objective is to harmonize UN capacities, mechanisms, roles, and responsibilities. And in the phase one of its work, what's been done so far is that there's been a mapping of biorisk expertise within the organization. There's been the creation of UN guidance on coordination in case of high impact disease outbreaks. And also, we've done some outreach strategy for the organization. In phase two, which is now emerging, we are going to work more on how we can have an exchange of knowledge and staff within the organization, and also making sure that biorisk management is reflected more centrally in the UN's broader crisis management framework. And continuing to work on outreach to relevant stakeholders, such as civil society, academia, industry. Again, here for us, this element of multi-stakeholder is very crucial. And I think if there's anything I want to emphasize, it is that message that the various actors increasingly I would say, need each other to be able to respond effectively to biorisk. Thank you.
Thank you so much. Multi-stakeholder, very important indeed. And we have a prime example of that on our panel here. Similarly, what you were saying about soft law and hard law needing to work hand in hand is a little bit similar to what you were saying this morning, David, about the need for both bottom-up and top-down processes. So we're already at this early stage in the conference hearing some recurring themes. So let's keep those in mind for our future discussions. Stephanie, can I turn to you to respond to how your organization is thinking about future biothreats?
Absolutely. I'll go back to my third point with regards to developing sustainable and competent professional networks in biosafety and biosecurity, and particularly focusing on commonly underrepresented stakeholder groups. I won't even just say participants, but key stakeholder groups in the global biosafety and biosecurity landscape, which is women and youth, but depending on regional context, can include a lot more, or many more different types of underrepresented populations. And before I speak about my organization, I'll kind of lead with a positive point about some other organizations doing really good work in this regard, emphasizing these populations' inclusion and having them be an example for responsible use with biological materials. One of them is UNODA's Youth for Biosecurity initiative, integrating youth that are interested in biosecurity that are in life sciences or policy spheres into basically collecting and disseminating key priorities that youth have for responsible use of science and technology moving forward. And I think this is a really great way of taking a global look at emerging priorities from youth as a stakeholder group in formal biosecurity fora. Another really good example of work being done that is kind of similar to or in line with IFBA's ethos on including underrepresented groups is iGEM's Human Practices program, which is under their kind of responsibility arm with safety and security. And essentially, the perspective there is to ensure that emerging projects that students create or that young people create in synthetic biology are responsible and that they equitably benefit all communities that may interact with it or that risk is mitigated in a similar way. A really, really nice example that they integrated into their programming for a responsibility conference that was held late last year was incorporating Indigenous perspectives on zoonoses, risks that are presented by emerging animal health threats. And so a lot of check marks are being made, and there is appetite for activities and I would say values or perspectives in terms of this growing responsibility or inclusivity in collectively assessing risk and being responsible for emerging risk. In terms of this inclusivity as it pertains to our own projects, one of the biggest points of feedback from our global membership was that biosafety and biosecurity, there needs more to be done to form a formal career path for biosecurity experts. Most of the world does not have a clear definition of how to become a biosafety or biosecurity professional, and it varies quite a bit from country to country. And one of the efforts with support from Global Affairs that IFPA is implementing is a dedicated undergraduate degree in biosafety and biosecurity in Kenya. And this project is well underway with a lot of support from stakeholder groups internationally as well as locally. And this will allow for the, I would say, legitimization of a lot of experts in the global sphere, that, as well as those that are up and coming, that may not have PhDs or dedicated degrees that will, give them access to formal spaces, but have years and years and years of professional practice and experience under their belt. Another point is formal research on diversity, equity, and inclusion in biosafety and biosecurity, and this is commonly viewed as adjacent to other topics in the biosecurity sphere, but I think it needs to be argued that it is critical to possible implementation or effective implementation over the long term. Not to beat a dead horse at this point, but going back to my borrowed analogy, if we don't have all of our little lights lined up on the runway, the plane might not see the end of the runway and might not take off, right? And so being able to have that robust collective perception of risk and diverse ideas on how to mitigate it is essential, and we need to include all of these underrepresented communities and professionals that reside within those communities in order to achieve this collective responsibility. And if we want to achieve collective responsibility, we have to acknowledge who is in that collective. Following that point, how do we ensure that there's progress in that regard, and how do we measure it? And so IFBA, again with support from Global Affairs Canada, is conducting some very exciting research on developing progress indicators in diversity, equity, inclusion that are specific to the practice of biosafety and biosecurity. And so I'm sure if you follow any of our communications, there'll be more to follow with that, but we're sure that will definitely shape how we view implementation of that stakeholder inclusion moving forward with evolving risks.
Thank you, Stephanie. Wonderful examples of some of that hard work and important work of actually building in stakeholder views. We all talk about the importance of multi-stakeholdership, and we all need those different perspectives included, but actually getting out there and doing it is the sort of thing that you're doing. So again, thank you for that. Sam, any thoughts about future biorisks at IEGBBR or perhaps in your national capacity?
Yeah, basically, I think I'm going to pick up from where I left off last time, where we spoke about capacity building. So moving forward, we are thinking about building more capacity internationally. So at this particular point, the whole idea is that we have 11 member countries that are involved, that are sharing knowledge and experience, but it's better to go beyond that. And so we are trying to get to the point where we can get developing countries involved in this kind of discussions, and also provide the expertise in those developing countries to be able to build, to strengthen the global biosecurity and biosafety oversight system. Remember that the world is connected, and as a result, anything that happens in one part of the world can affect other parts of the world, and therefore working together, not only for those 11 countries, but for other countries will be very critical in addressing this challenge in the future. So that's the first thing we are trying to think about, do that. So as I said earlier, we've already been in contact, thanks to CAC, we've already been in contact with Africa CDC to extend our expertise in terms of what is needed in order to build that particular capacity. We've been able to also document all the expertise that we have in the 11 member states that can be helpful to developing countries in actually building or strengthening their oversight capacities. That is the first thing we are hoping to do, and if we continue to get more funding, then we're going to extend it beyond CDC, Africa CDC, to other parts of the world, that South America and also Central America and also Asia Pacific is what we are also looking at in the future. Secondly, in the past, we used to have meetings with 11 member states. Again, in order to extend the capacity, we're trying to also invite other countries to be part of these particular meetings so that we can always discuss and share knowledge across the bigger group of people. So that's, again, another kind of activity we are hoping to undertake moving forward into the future. And then at this particular point, we also think about reporting surveillance systems as very important in order to mitigate risk. And so basically, at this particular point, we are working on projects that will actually bring together all the surveillance systems that exist in all these countries so that we can be able to share and also make sure that other countries can be able to use that to develop their own surveillance systems. So I'm talking about lab incidents notification systems that will help you actually try to determine what is happening and how to mitigate that, reduce those incident exposure and non-exposure incidents in those labs. And so there's a work going on, and that we expect that the output will come out by the end of August, where we are going to put together all the experience and surveillance system that exists in these 11 member states together as a capacity building tool that can be used for other countries to develop their own systems as well. So in the nutshell, those are the three items we are looking for. Remember, IGDBR depends on funding, so our outcomes are quite short term. It's not going to be into the next 2 years, but within this particular fiscal year I'm talking about at this particular time.
Thank you, and it's really an important point, this point about funding, because we cannot do what we want to do without funding, and a number of you have already mentioned by name Global Affairs Canada, which is an incredibly important supporter in terms of global biorisk management, also supporting work at the WHO, at the UNODA. So funding, of course, is key to a lot of this. And tomorrow there will be a panel from Global Affairs Canada, where Global Affairs Canada is represented, and where we'll hear much more about their initiatives. The private sector, of course, is a different source of funding, and I think an important one, and one we've really not drawn on so well when it comes to biorisk management. Can you tell us a little bit, Alex, perhaps not so much on the funding side, but on future biorisks and the sort of work that you foresee for perhaps Blue Dot, but more generally the private sector?
To be clear, we are very interested in funding. But yeah, I mean, I'm mostly excited. So part of this is just me as, nerdy data scientist, but all the information sources that have come online in COVID, investments in new detection technologies, international deployments, more awareness and more general information sharing is really exciting. I think there's a real opportunity to get better synthesis, earlier, faster, richer assessment of events. It's just purely exciting as someone in this field for a long time. And then in parallel, a revolution in cognitive technologies like large language models. Scary elements, but also really exciting elements for useful upgrades for our public health and biodefense capabilities. I'm also excited for a bit of a lull in the, say, wartime discourse, particularly around interpretation of evidence. I mean, a lot of our job at Blue Dot is synthesizing, collecting information, but also translating it effectively for action. In a context where evidentiary standards and how to interpret this information is being worked out in real time, and it's a contested, somewhat adversarial information environment, it's next to impossible. So I'm pretty excited for a lull from the public health crisis and the opportunity to settle on some standards that we agree to call our shots in advance, and be able, next time something of this nature occurs, have more standardized methods that are pre-agreed to that we can use to reach quick judgments. So overall, pretty excited about the future.
That's what we want, excitement. I'm conscious of the time, and I really want to get a sense of the temperature of the room, some points from all of you. So what I'm going to do is to collect a whole range of questions, comments. Please keep them short. Please do introduce yourself. We've got about, I'm going to take an extra 5 minutes, so we've got about 8 minutes or so to collect your points, and I see there are already many in the room. So we'll start over on this side. If you briefly introduce yourself and try to keep your remarks as short as possible.
Thank you. I'm Florence Desbarres. My question is for Alex. You mentioned Blue Dot's achievement, that you were among the first to pick up a signal of something going on in Wuhan. You mentioned a business website, and I think it's a Finance China website. And then I checked that it was mentioned it was on December 30th. So my question is, did you try to retrospectively look for earlier signals? If so, did you find any? And if not, was December 30th the earlier signal your top-notch technology could pick
that's a great question. I think
Alex, we're going to hold. We're going to collect questions. Bear it in mind. Thank you. Next question will go to the back, in the middle.
Yeah. Hi, my name is Paula Dupalibias with Geneva. Yeah, okay. Geneva Solutions. My question is actually for the WHO representative, considering all the discussions that, the considerations that have been mentioned, ethical, these security considerations and so forth, I was wondering, with the pandemic treaty in development right now, how much are these considerations going to be part of the treaty? Given the concerns about the possible leaks, lab leaks that have taken place, whether these issues are being discussed within the context of the treaty discussions.
Thank you so much. I'll take David just next to her.
Hi, David Gillum, past president of the American Biological Safety Association, and work for Arizona State University as the assistant vice president for environmental health and safety. As the previous panel mentioned, and as people on this panel have mentioned, the rules are different everywhere you go. And there's quite often a disconnect between those creating the biosecurity policies that are out there and those who are implementing them. How do you feel that we should address that gap between the policy making and the people who are actually implementing?
Important point. Thank you, David. We got one in the second row.
My name is Jonathan Latham, and I'm a virologist, and I have some sort of brief comments, really, rather than a question. But firstly, I wanted to point out that we've left an article on the table, which is an Ebola outbreak assessment of the scientific evidence for whether it was a lab leak or not. So I just wanted to point that out because it's here to share, and I bought more copies that I can I can print more copies too. So, but I wanted to just share some personal experiences because...
shortly, though.
Yeah, so they're quite brief. so the issue is that, you know, I have had experience with projects to improve and reform scientific and regulatory systems, for example, in toxic chemicals. And, you know, we're here to improve the regulatory systems around biosafety. And what I've, you know, the sort of consensus around successful efforts to do this is that there tends to be an outside strategy and an inside strategy, right? There's a double strategy. If you talk to campaigners who do this kind of thing as a professional job, and these double strategies they're not necessarily coordinated or deliberate, it's just something that tends to happen. And the characteristics of the inside strategy is it's nuanced. You know, there are people who understand the scientific niceties, the organizational opportunities, the legal situation, and so forth. And then you have a bunch of people on the outside who are quite loud and quite noisy, and they might wear J.O. Fauci t-shirts and so on and so forth. And there's a tendency between people on the inside and people on the outside who are exactly the same objectives to see these two strategies as in opposition to each other. And the understanding of campaigners is these strategies are not oppositional. They are complementary to each other. Right? You can think of them as hammers and chisels. Right? You need a hammer is no use without a chisel, and a chisel is no use without a hammer. And scientists, in my experience, have a very high opinion of their own abilities to make arguments on the insides of discussions, and in, you know, confidence in their ethical positions that these things will make a difference. But the experience is that it only makes a difference if you can have a noisy crowd outside the room who are making the policymakers, the establishment, if you like, listen to them, right? So the issue then becomes, you know, how to coordinate between these two groups. Because the inside group, you know, there are class differences, there are all kinds of issues between the inside group and the outside group.
Jonathan, I'm going to stop you there, but I think your point is well taken, that there's a role for different kinds of activism in our society, and these groups need to speak to each other. We have very little time, and there's a couple of more points that I want to get in before we wrap up. So if there's a final point you want to make?
Well, yeah, I'm sorry if this is boring, Philippa, but the issue is that these two groups, you know, there are essentially, frequently misunderstandings occur between these groups, which I've seen happen in this case, right? And what happens is that the people on the inside have a lack of appreciation for the people who exist on the outside. They often are morally clouded, whereas the people on the outside have a certain kind of wisdom that comes with, you know, they're angry. We're angry about, you know, not me personally. I have a certain level of anger, but you know, we've had a pandemic in which 18 million people have died, countless lives have been ruined, and so on and so forth, and yet we have a conversation on the podium about GoFrog, right, which is basically a mealy-mouthed definition of risky virus research that I personally would bet my house was the result of some kind of a lab accident. And yet the people on the outside are perceived as not having a strong moral position because they're perceived as not understanding the issues, whereas GoFrog has no real definition. It's a totally faux scientific term that is basically being used by the establishment to divide those two groups, right? So the two groups that work together are being divided by a silly set of terminologies, which makes it difficult for those two groups to talk to each other.
Okay,
so
thank you. But we understand your point. We have another day and a half of conference to continue this conversation. So please let us proceed with the panel. And thank you very much. I'd like to go to Jerry at the very end.
Thank you. And real quick, Jerry Parker, Texas A&M University. So the question is for the NGOs and the private sector on the panel. So in addition to funding, what can national governments do to better incentivize your very important work. I'm very excited about the work that you presented today, and not only national governments, but the WHO and the United Nations. That's my question. Thank you.
Thank you so much, Terry. We have a final question at the front, and then I'll turn to the panel for any closing remarks that you may have. Just here.
Thank you. My name is Shahid Jamil. I'm from Oxford. Question to something what Alex said about technology. You cited wastewater surveillance. Technology has done really well during the pandemic. Especially now we see how wastewater surveillance has been useful. But I think it's also important that during this period of lull, we don't become complacent and we continue to do the surveillance. Unless you have a baseline, you don't know when your spikes come. And so that's the one point I want to say.
Thank you so much. Thank you for all very useful comments. We have taken note of them all. I thought we might go in reverse order this time. Alex, if you're happy to kick off.
Sure. Well, I'll speak to the super interesting question about earlier signs. That's always fun after an event. So if I have the timelines right, 2 or 3 weeks before the signal we recovered, there was mostly social media chatter on Chinese channels that we weren't monitoring, and I don't think many folks were. A lot of those are closed WeChat small groups that are really challenging to access, but there was some secondary chatter on Xinbo. I think some other Chinese social media that I think got into local reporting as well. So there's a potential signal there. The other that's a bit hard to imagine scaling to online global monitoring is an uptick in hospital parking lot usage detected by satellite imagery early-ish December. This is in a class of post hoc detectable signals that are challenging to imagine using prospectively to detect threats, but that maybe is the theoretically earliest detectable sign that I'm aware of.
Thanks, Alex. Sam.
Yeah, so basically I'm just going to pick up on the question over here or comment that was provided. I think the key message here is risk communication, which has to happen no matter what the problem is. I think for us in Canada, I'm going to put on a hat as a regulator in Canada. We do tend to communicate a lot. There's no confusion between the stakeholders and the regulators. Basically, what we do is provide regular webinars to our scientists and regulators to understand what has to be done to reduce the risk. You know that scientists are not there to cause problems. We also believe that innovation is important, and therefore we are not going to restrict that as well. So again, as a regulator, if you put more regulations, you are damned. If you put less regulations, you are damned. So you have to find a middle ground where you can be able to operate, and we believe that communication is the key. And so we do develop a lot of webinars where we talk to our stakeholders to understand the risk that is involved in their research. We do provide a lot of what we call the safeguarding science webinars that will talk about dual use and life science risk on a regular basis. And so that is what I think is going to be important to get the two to come together so that everybody will understand what is required in terms of mitigating the risk. And basically, I think that is something that has to be done. Again, as a regulator, we are not here to actually restrict innovation. We believe that is important, and many scientists understand that as well. Just to tell you a little bit story, recently I have a scientist, a researcher coming to us, talking about a particular sequencing that they want to do and wanting to know whether it's a gain of function, because it's important for them to understand that. Initially, he didn't know it was gain of function. He had argued that research is not gain of function. And so basically when he started, I asked the question, what result are you expecting from this outcome research? And he said, well, we expect to have the same kind of pathogen, but we don't know. Something can happen. We can't control that. And I stopped him to say, right there, you don't know what is going to happen, what the outcome will be, and that is telling you that it's a gain of function there that has to be taken care of. And as a result, there was an understanding that that particular research was gain of function, and certain mechanisms have to be put in place to mitigate the risk. So again, communication is key in terms of all these activities to make sure that the two parties are talking the same language Otherwise, things are not going to work out properly.
Thank you, Sam. Talking the same language can often be harder, easier said than done. Stephanie, any brief final remarks?
Absolutely. I will address David's lovely question with regards to policy implementation, especially translating international standards or national standards that may be viewed as better or best practices to other countries. And I mean, I am biased coming from IFBA, but biosafety associations are very, very well positioned to do this, especially considering many of their membership are well placed across government, industry, academia, and other nonprofits, where they can translate these best practices into local contexts that can be implemented over the long term. Another thing that I would also mention in terms of avoiding this policy implementation gap is ensuring that in the formation of these best practices or guiding documents that you do have robust stakeholder consultations, ensuring that, you know, in terms of geographic region, industry, and other stakeholder groups that I've mentioned today are all able to meaningfully implement or contribute to following through with those practices. Another question that came from across the room here from my colleague about how IROs and national governments can best support NGOs. I can't speak for private industry, but I would say in terms of a good example, Public Health Agency of Canada has been very good to, I would say, lower level stakeholder groups. A good example would be an outreach event they held actually shortly before the pandemic started on synthetic biologists local to Ottawa and regionally as well. Sharing their perspectives, what people who are working on the front lines or in community groups encounter in terms of barriers or opportunities. Another good example is they were present at Youth for Biosecurity's very first workshop held back in 2019. And so if governments have human capacity to spare, having representatives on the margins of formal meetings attending these side events or workshops as experts is really valuable, and I think raises a certain amount of awareness for emerging priorities, such as youth or community consultations. And a final note, time is up. I will just say that there is a similar chisel and hammer argument with gender and disarmament. And I think while it is good to advocate for these stakeholder groups for advocacy's sake, it is also interesting to further develop an evidence base formally and see it as a question of efficiency and ensuring that we don't miss the same blind spots in terms of stakeholder engagement and who is handling these biological materials day to day. So thank you very much.
Thank you, Stephanie. Ade, do you have any final 30-second remarks?
30 seconds, I was just warming up. Now, I will say two quick things. One was the last question that was posed from there. And to say that from what I pick up sometimes, it seems that in terms of some national governments, within those national contexts, you know, I was referring to COVID as a wake-up call. Some, to be careful, political constituencies within those countries still seem to think that when the rain falls, it doesn't fall on their house. I think there's a need for national government, by which I mean those who are well-resourced, to realize that pathogens are not bullets. You know, so you can, when with biorisks, it is a universal risk. So funding capacity building should be universally directed. Otherwise, it's going to come back to you. First point. Second and the last point is actually directed towards us and back to my ventriloquists. We have the emergency platform. We have the UN Biorisk Working Group, which we are using as instrument for this multi-stakeholder approach, I think the homework we have, and we are inviting all of you here, is to try to build that bridge between the United Nations and all other stakeholders, so that when there is the next, God forbid, any such bio incident, the United Nations is able to know who to call on, how to call on them, where they are located, so that this platform is able to function. So we are inviting you all to operationalize the multi-stakeholder approach of the UN. Thank you.
Thank you so much. Emmanuel, you have the final word.
Thank you. So perhaps, do you want me to just to answer some of the points or just to provide final words?
Just final words, I think, at this point.
Thank you. So I'd like also to just to re-emphasize some of the point that has been made here in the panel, but then also that have been made also in the presentations, that it is, biorisk management is a global issue impacting all countries. And so we have heard there's no one-size-fits-all, there's not a single approach. And so again, we've heard also this notion about shared responsibilities, and this is also something that we have been taking during the development also of the framework, but then now in the implementation to have this multi-stakeholder approach, as it has been mentioned. And so I think I will end with this note to continue to engage, collaborate, and to find synergies and create them.
Thank you so much. Friends, colleagues, ladies, gentlemen, you have been very patient, but I hope this panel has given you some insight into the very active work that is going on globally when it comes to biorisk management. For myself, I think my key takeaway is that there is still scope for some collective strategizing between all these institutions, bodies, networks, and to draw on these synergies, as you were saying, Emmanuel. Before we thank our panelists, I would like to note that we're running a tiny bit late. We will reconvene again at 4:30, so you have a few minutes to stretch your legs and have a comfort break. But please do come back promptly by 4:30 because we have another fantastic panel lined up for you. So with that, again, thank you for your attention, and please join me in thanking our panelists.
Welcome back. My name is David Heyman, and I am a professor at the London School of Hygiene and Tropical Medicine. And I'm really pleased to be chairing this panel at this point in time, because what we do is go from the level of pathogens to the level of global mechanisms that are ready to support countries in their biosafety. And now we're going actually to the country level. And today we'll hear from two different parts of the world. Tomorrow you'll hear from a couple more. So today we'll hear from two excellent panelists. And there's good news and bad news about this panel. The good news is that the panelists are excellent. The bad news is that this will go on till 5:15, but we will turn that bad into good, as you'll see as we continue. So let's get started. And I'd just like to start by asking each of our panelists, maybe Nasreen first, to introduce herself, tell us a little bit about her background, and also about what she sees as the major biosafety hazards or biorisks in Jordan in this case. Nasreen.
Thank you very much, David, and good afternoon. I'm Nasreen Lahmoud. Microbiologist, bioeducation. I'm the director of the Biosafety and Biosecurity Center at the Royal Scientific Society of Jordan. I've been involved in issues related to biosafety, biosecurity on a national level and regional level in the Middle East and North Africa for the last 15, 17 years. And I've been involved in different and several networks in order to have, to build the capacity of regional champions in issues related to biosafety, biosecurity, dual use, other, issues related to pathogens, in the labs and how to handle them. We don't have, the major challenge I will speak about, for the last 15, 17 years, we tried to establish several networks, several associations related to biosafety, biorisk management. However, and these networks are very important because they are like a mechanism and a tool for integration and coordination among different stakeholders and champions in the region to build their capacities and also to enhance ownership of these training courses and capacity building activities. However, the problem was to sustain these networks, to sustain these associations. This is a major challenge that we are facing in our region.
Thanks very much. So sustainability, a very important issue in your networks. Anurag, tell us a little bit about yourself.
Sure. Pleasure to be here. By background, I'm a physician scientist, trained in pulmonary and critical care, until recently ran India's main lab in genomics. And I'm currently Dean at a new school of biosciences at Ashoka University. My exposure to this area came from building a biosafety lab in my former institution about 15 years ago, and the biggest challenge that I see is the challenge that has arisen out of opportunity. India has about 20 biosafety 3 labs, a lot of them focused on tuberculosis research. We have at least 2 biosafety 4 maximum containment labs. 2 more are under planning. And what I see as the biggest challenge is that most of them get created for research and are expected to be sustained by research. They require a certain amount of manpower to do them very well. The infrastructure, the engineering when it comes up is state of art. The people who are the PIs are well trained, coming from the best places in the world. But when it comes to day-to-day operation, it's really about brass tacks. It's really about finding the funding and the people, and finding the funding and the people in the context of what is permitted by the rules. This, to me, is the biggest challenge.
Okay, so you're constrained by personnel and you need more personnel, and those personnel who you take may not have the training necessary to deal with the biohazards. So let me push you a little bit further and ask you what the solution is to that.
Well, there are 2 parts to it. First, of course, is training of new people as they come in. And that's usually a worked out problem. Most people have a protocol and SOP that a new person can be trained in. The challenges come from 2 directions. First, the researchers themselves, who have a project in mind and are in a hurry to push and complete it. So they are looking for shortcuts in getting their people into the lab. You get a fresh PhD student. There is a project they need to do. You want to find a way to get the guy who runs the biosafety operations to quickly let him in. The second is about retaining people. Typically, at least in my country and possibly in many others, the value is in the degree, not always in the experience. So a high-level technical person whom you have trained but may not have the PhD degree will cap out at a particular point, at least in those sectors where the funding is written into the grants. And then you lose these people because they will move on. Of course, the private sector gains, but then many of the research labs end up as a place with a continuous merry-go-round of new people coming in. And at some point, shortcuts become part of the game. So only those labs that are built for purpose, a purpose in which there is a clear national, well-established system, sustain. Others keep suffering this recurring manpower problem, particularly universities.
Thanks, Anurag. Nesreen, let's come back to you.
Yeah.
Your networks that you're trying to sustain, is there a solution to that? And do you also have this same problem of laboratories that are well supplied at one point and then after the money dries up, they're no longer?
Actually, this point is valid also in Jordan. Regarding biosafety and high containment lab, biosafety level 3 lab, actually we have one in Jordan, and it's really very difficult to operate and keep it sustained and maintained. So even if you have the initial fund in order to have this type of high containment lab, how to sustain the operation in this lab is a challenge for countries like Jordan or other countries in the region. For example, we have some labs, for example, in North Africa, in Morocco, they are well established and operated because I think they do have joint collaboration with African countries. So I think they considered as a reference labs in order to test infectious disease in their samples. So the situation, even in the same region, in the Middle East and North Africa, maybe it differs from one country to another country. How to sustain networks is really very difficult. Maybe we need to have the culture among our staff and technicians in the lab, among the professors and the students in universities. Recently, we have joint collaboration with the Sandy National Labs, the US Sandy National Labs, in order to have a curriculum for risk management, for students, because we didn't have such a curriculum, at universities in Jordan, and even such a curriculum is not found in many countries in the region. So we need to have this, baseline and this culture, and to build it, from schools to university, in order to sustain the activities and the capacity building that are currently running in the region. Capacity building activities and training courses and the funding for the region started, I think, 2008, 2009. And a lot of activities and training courses have been implemented in the region. However, if you want to see, the implication of all these training courses and activities that have been, conducted in the region, I don't think that it's more than, more than 20%, 30%, we can say that, it was successful because we couldn't sustain these efforts. Because we don't maybe establish a culture among our technicians related to biosafety and biosecurity.
Also, Nesreen, we heard earlier that regulation is difficult because it's hard to keep regulation up with changes in laboratories.
Yeah.
What about regulation in Jordan? How's the regulation?
Yeah, it's not only in Jordan, also in the whole region, Years ago, with my colleague, Mr. Tim Triple, we tried to have this network, the BBIC, Biosecurity and Biosafety International Consortium. And one of the activities that we were aiming to have a federation of association of biosafety, and then we have, national and regional regulation. In Jordan, we have the National Biosafety and Biosecurity Committee. It was recently developed, but it's not really active. And this, we have the same issues in the region. We have some guidelines, some SOPs in some universities, in some research institutions. However, to have a national regulation like the situation in Europe or in Germany, we don't have this, to be honest. So it's really, we can't compare the situation in our region with what's happening in the US or in Europe.
Anurag, how about in India, regulation?
I think I'm going to borrow what Gustavo said in the opening thing. I think we all know what to do. The rules are very much in place. The difficulty is in translating that knowledge into action and doing it professionally. I think that word that you used, Gustavo, was one of the most important. The regulatory part, at least in India, is reasonably well developed from the levels of guidelines from the top authorities. Even the systems of regular implementation, checks of biosafety facilities are all there. It is that soft money and soft job problem that leads to trouble. And I think it's probably not just India, it is probably all over the world. Because of research, we have started using soft money and soft jobs to do this mission critical work, which probably was not designed for this amount of last minute thinking about money and people. And that's where I think in India the regulations exist, but may not, particularly on the academic side, always translate to on-ground action. I'm reasonably certain, without knowing, that the biosafety 4-level lab run by the defense labs of India are probably well managed, because they would probably have a permanent job in the government for every person who works in it. At the same time, in my institution, where I create soft jobs for people working, it's a bit more of a challenge. So I think the challenge is not so much in knowing the regulatory side, the challenge is on implementing the regulatory side.
Nasreen, yeah.
I think, we don't have, like a national regulation, but, like it differs from private sector and public sector. For the professors in university, they don't have this awareness regarding biosafety and biosecurity and the regulation, and even the BTWC. We have this project funded by the EU years ago, and we want to see and test the awareness among professors in university regarding different issues, biosafety, biosecurity, dual use, and unfortunately they were not aware about these issues, or even what's BTWC and this thing. And because of this, we had this national regional workshop that we conducted in order to make this awareness among scientists and professors in universities about issues related to BTWC and advances in science and the impact of advances in science might have on like dual use issues and that things. For the private sector, and maybe they do have like internal guidelines, internal SOPs that they follow, and it's related in line with the international regulations. So there are following this regulation. Maybe the situation differs even in the same country from one lab to another lab, from one facility to another facility.
So there's no general accountability mechanism that exists?
No, no.
Should there be one?
Yeah, there should be one, and I think because we recently established the National Biosafety and Biosecurity Committee, and it includes stakeholders from different ministries in Jordan, so maybe they will have the tool in order to audit different labs and how they handle pathogens and different biological agents.
Anurag, accountability mechanisms in India?
Not very strong, because there is no teeth to the regulators per se. So at most, they are not required to have an audit done by a particular agency with teeth to enforce anything. So yeah, that's really where the problem is. No regulator with real
Oversight,
teeth
that
can provide genuine top-end oversight, with the ability to shut down a place that was found to be not meeting up to the standards. Sometimes I wonder if it is by design that we as researchers don't create those mechanisms. But yeah.
What about the WHO and the Global Health Security Initiative, or agenda rather? What about their joint external evaluation? Nasreen, have you had a joint external evaluation, and has that done anything to look at biosecurity?
Yeah, we had this, I think, in 2016, and I was involved in this, mission. There's a part for biosafety and biosecurity and how, like to assess the situation in the labs. And I think on the website, they publish the national reports based on this, assessment and JAE visit. Yeah, it's part of the mission there.
Yeah. Did India do a JE? Do you know, Anurag?
I'm not certain. I don't think there is one place that comes to do.
Mm-hmm.
More often than not, it might be a reputed company that comes and provides a certificate of operations. We know how that has been working in the finance world, hasn't we? So it's probably working along the same directions. The people that you pay to keep your systems running are the ones who do the audits and come and say, yes, it is running well. That has a point of failure because of conflict.
Let me come back to you, Anurag. You said earlier that soft money was a problem in making sure that there's good biosafety and in continuity in research. What about the laboratory equipment and the laboratory systems for security? Are they impacted by soft money as well?
Yes, they are actually, because if you look at any good lab, you have to pay maintenance contracts. And all those maintenance contracts are typically a percentage of the initial value of what you buy. And this differs tremendously between, say, a biosafety 2 lab and a 3 lab. In a 2 lab, a country like India that also produces equipment can keep the initial cost down low enough that long-term operation is very sustainable. While the minute you go to higher-end containment facilities, 3 and 4, a lot of this stuff is imported. And when things are imported, your running costs are automatically high. And the other thing that happens is that as the country, in our case, goes through an indigenization drive, which says anything that is locally produced would be preferred, and the path towards bringing outside items becomes more difficult. There are huge operational challenges in keeping this, both from a money point of view, because almost every researcher says, give us the money, we'll build this facility. In 5 years, we'll sustain it ourselves. Yeah. And that doesn't quite work out as well.
Thanks. Well, let's go down to BSL 2 level and maybe even lower. And let me just pose a not a hypothetical problem, a real problem today. As you know, there are many laboratories in the world that have specimens, stool specimens on storage from times when they were collected during polio, when wild polio virus was present and when Sabin virus was present. Those are stored in laboratories, and they are taken out from time to time, not necessarily in a BSL-2, maybe even in a lower laboratory. The question is, are you ready for those? Is your biosafety and biosecurity in Jordan ready for that study, should those potentially infectious materials for poliovirus be used?
I think, we have at the Ministry of Health, Central Public Health Labs, at the Ministry of Health in Jordan, they have the requirement in order to handle this type of samples. And I think they have the infrastructure, and also we have biosafety level 3 in Jordan. So if anything happened and they need to conduct any research on these samples, they are capable of doing this. For our labs in my center, we are working with, a project on, surveillance for coronaviruses with EcoHealth Alliance in New York, and we are a reference lab for, regional, in the region in, Western Asia. So we'll collect, and we did surveillance in Jordan and in different countries in the region, and we collected a good number and, hundreds of samples, and we have the capability in order to store these samples in a safe and secure manner.
Right. Very interesting question. I was thinking about a bit more while Nasreen was speaking. So I think the official places that store these things, whether rotavirus, whether polio, are probably quite safe, because these are designated hard money places. But there are plenty of clinical places in India that might be doing simple research. I mean, for us, rotavirus or polio is not something that is never seen. So perhaps the gravity with which these things are taken may not exist in the routine clinical environment which sees these people. So would people potentially have samples from patients in their freezers without adequate biosafety facilities? Quite possible. I would not say that I know, but imagining the clinical facilities, the usual situations, the usual places where these people really come, I would not be surprised if those were potential points of leak. But no, the national labs at this level would be good enough.
Yeah, okay. Okay, well, I think it's really important that we keep the focus on all laboratories and not just on those that handle the exotic specimens worldwide, which is what many times we focus upon. And I think those are good points. In academia, you're quite free compared to what happens in government laboratories.
Yes.
And that's a good. How about in the Middle East? Is that the same?
They're the same, yeah. I think, you know, like if these samples will be stored in like a governmental lab, it is, you know, well maintained and secure. But related to the academic sector and the researchers, what they are doing in these labs, there is no mechanism to, you know, oversight these activities.
Nasreen, you're a champion for biosafety and biosecurity in Jordan. Are there champions in other parts of the Middle East?
Yeah, there are different champions. And, you know, like I mentioned earlier, that we try to establish several networks in the last 15 years. And although these networks were hard to be sustained, we are still coordinating our activities with, you know, the champions in different countries in the region. Yeah. We conduct, you know, capacity building activities. We try to, like, you know, develop guidelines, you know, a platform in order to share best practices about what's happening in our labs.
Anurag.
So we have official departments that come up with guidelines for the training and everything else. So that part's good, but I don't know of people who are looking into the nitty-gritties across the country in a cohesive way. That could probably use improvement.
Yeah. We've heard a lot about the extraordinary global resources that are available for countries. We heard that in the previous panel, and it's quite impressive. And I just wonder if you've come into contact with any of these during your routine work, and if not, how they could be better advertised and maybe operationalized in your countries. Nasreen, are you aware of all these different networks?
You know,
You,
like
of course are, yeah.
In the last two panels, they speak about, you know, like, you know, WHO and the UN mechanism and all these things. And, you know, in Jordan, you know, we get a lot of funds from different agencies, international, collaborative international funding agencies. So, you know, like, you know, 2008, 2009, funding for the region and for Jordan started in different domains related to biological agents and pathogens. The focus were mainly in the military sector and in the public health sector. And then, you know, it goes to the agriculture sector. And recently, they started, you know, doing some funded projects in the academic sector. Because of this, you know, the professor in university and students are not like fully aware about these issues. But we are trying, you know, in order to enhance and develop the culture among professors and universities.
Anurag, has it reached the academic level in India?
Very much so, but you know, to be very honest, when I hear about lots of money becoming available, I worry more than I become happy because I see what's going to happen. I see people will upgrade a BSL 3 to a 4, a 2 to a 3, and this is sometimes short-term interest. My own institute, when money became available, built 2 BSL 3s because money was available. That's the nature of what we do. But when this money will dry up, nobody gives you the permanent path, and these things need to be built for the long term. So I think short-term money becoming available is usually more of a trouble down the line than a blessing. So it's one of those interesting things. One must be very careful how one sees it.
Okay, thanks. Well, I'm going to open it up to some questions from the audience, and then we'll come back to the panel. But does anybody have any questions? And I'll take all of them at the same time so we can get a whole series of questions out. There's one in the back, yeah.
Thank you for a great presentation. As Nasreen knows, I do a fair bit of work in Pakistan, and many of the problems you both talked about I see pretty much around the world. The issue of old samples is everywhere, including in Europe and North America. But one of the problems that they have in Pakistan, and I'm really interested to know if you have the same problems in Jordan and India, goes beyond the soft workers you're talking about that you yourself employ, and goes to the external professional ancillary people you need, like there's one company in Pakistan that has the capability to certify biosafety cabinets as being operational. One company. there are no architects in Pakistan who can design a BSL-3 lab. Is that the sort of problem you have in India and Jordan? Thanks.
Okay. Other questions? Yeah, in the front here. Okay. Yeah, and then we'll come to you.
David Gillum again. Thank you for sharing your experiences. I think it's really valuable to hear these international perspectives. I just want to make a comment. So I did participate in a Sandia twinning event with a colleague, Gaia Awadani from Jordan. I heard she just retired. I'm really sad about that. But what can you possibly talk about other collaborations that might be beneficial internationally besides that or in addition to that?
Thanks. And in the front here.
Thank you. Mark Lipsitch from Harvard School of Public Health. I'm curious, one of the good things about this gathering is the larger number of countries represented than in most of these discussions. And I'm curious from each of your perspectives, which haven't been very much evident in the discussion over sort of pandemic risk research, research that risks creating a pandemic pathogen. To hear your thoughts on that in general and what are how you see the risks and benefits. Much of it has been done either in China or in Western countries, so I would like to hear more perspectives.
Okay, any other questions at this point? Okay, let's turn back to the panel then, and go ahead and answer whichever questions you feel you can answer. And we'll come back.
Let me take the first one that's easiest to answer. No, we have more than one, but still a small number. So it's still a small number. So at the time, for example, when we were building our BSL-3 labs, there were 3 people in India capable of designing. I'm sure the number is significantly greater by now. But getting the same set of people to certify that you otherwise hire is a common problem, and that's why I was alluding to the finance industry for the problems they face in their audits. So yes, I see that. The larger perspectives about global research perhaps can wait. The Sandia question was important. I myself went to Sandia Labs when I was the biosafety officer in charge of creating a biosafety labs. That effort is continuing in India, and I believe many good people have been trained in interactions with Sandia. I truly believe that US, India, Europe, India, international collaboration for training of people is not where the limitation is.
Nesreen.
Yeah, thank you, Tim. Actually, in years ago, we had, like a paper that was published, and at that time we did a surveillance for, different countries in the region, and also like in interviews with the experts in the region regarding the gaps in issues in biosafety and biosecurity and biorisk management. And the point raised by Tim, regarding we do have like, you don't have the skills and expertise in professions and in, technicians who will be able in order to do, for example, lab design or operating the lab or, engineering control. These, professions are lacking in the region. So if we want to to establish or design a lab, then we need to have the expertise from abroad. We don't have, in Jordan or even, in the Middle East and North Africa. Because of this, one of the projects that we were aiming to do years ago with Tim in the BBIC, how to design, a lab in a low-resource countries and with harsh conditions. So I think this is a good idea that not all countries can design, high-containment lab, or, if you want to do any research on, dangerous pathogens, and we don't have the special and the specific lab in order to do this research, we need to think about, designing similar labs which can, where we can operate and handle pathogens in these countries. At the same time, securing, the lab environment and, making sure that all requirements regarding safety and security are established. So we'll have, this lab design according to the situation in our region and different countries, where sometimes, we have a limitation in water resources. energy supplies, these things, also harsh climate conditions, hot weather, dusty conditions. So we need to think about these solutions in our region, how to design labs in line with the conditions in our region. Right. Regarding, Sandia, our collaboration with Sandia years ago, and not only the Ministry of Health, also RSS have collaboration with Sandia National Labs. And in 2018, and with our collaborator at Sandia National Lab, we developed the curriculum for universities, for professors, and it was the first curriculum to be developed for Jordanian university. And now, it's like many professors, take this textbook and teach it at universities.
Let me turn to veterinary laboratories. We heard this morning from earlier this afternoon about veterinary laboratories and the importance of research in the veterinary area. In India, are there similar controls or similar discussions about veterinary labs as opposed to laboratories that are mainly dealing with human pathogens or with humans?
I will have to confess ignorance.
Sorry?
I'll have to confess ignorance to that question. I'm sure there might be, but I'm not aware of it. And in fact, the fact I'm not aware of it makes me think I need to go back and do better, because clearly with One Health, I should have been thinking about it. But no, I kind of...
None of us think about it. That's the problem. We think only about what we know best. And it's always been said that the veterinarians are the ones who are more tuned to One Health than the human side. And I think that's probably true.
I think Shahid may have an answer to that one.
So one of the BSL-4 labs in India, one of the 2 BSL-4 labs, is in the animal disease domain. This is the one in Bhopal.
Yeah.
But yes, the concern is again labs that are there in agriculture universities, in the veterinary colleges. There it's as bad or as good as anywhere else. Yeah, and to add to something that you said about soft jobs and soft money causing a lot of heartache, so to say, this problem is prevalent in the region. And the final thought about your comment on BSL-3 lab designers in the country. India has been fortunate to have a very old space program. So there were people who knew how to make clean rooms to a very high standard. Some of them have repurposed to also build BSL-3 labs. So we are a bit lucky in that sense. But yes, I think the point about the same people designing and the same people auditing is something to worry about.
Yeah. Thank you. We also heard today about the importance of environmental surveillance, how, and that's been true in the Netherlands, but also in the UK, finding polio, and in New York, finding polio, in Canada. What is the role of environmental surveillance in laboratory safety?
Yeah. In Jordan, we are good in doing this surveillance for wastewater. And I think after the COVID-19, we are currently doing surveillance for wastewater in wastewater treatment plants for the effluent and influent in order to see there are, SARS-CoV-2 or any other pathogens. So we do this surveillance on a routine basis.
In the laboratory?
Yeah, so like, Ministry of Environment with the Ministry of Health, they coordinate their activities. Regarding, your previous question related to, before the focus was on Ministry of Health or the funding, in order to enhance the capacity building for, the technicians there and the staff there. And then recently they want to, they focus on One Health approach. So, they want to raise the capacity among, different technicians in different domains. So you can find someone from the Ministry of Health, Ministry of Agriculture, from the professor at universities, from sort of environment in order, to raise the awareness and build the culture among different sectors in the country.
Thanks.
I can't think I can say that India is good with environmental surveillance, but the science of environmental surveillance is well developed. There are people with expertise. One challenge for us that I've seen is everything comes under a different ministry. If you wanted to go check wildlife, you would need to go to a different ministry. You wanted to go towards sampling sewers or the water plants, you need to go somewhere else. Recently, India has created a One Health Institute, which for the first time will not be under one ministry, but under multiple ministries to make getting permissions easier. It's being monitored at the highest level, but I would yet to see how it goes. In terms of the science, it is good. There are people with genomics expertise, sequencing expertise, even in SARS-CoV-2, sewage sampling for viral load, sewage sampling for sequencing, and detection of new mutations in the sewage samples has been started. Even before this was happening for flu, it was happening for polio. And even environmental samples, for example, as hospital effluents for AMR, even these things are occurring. But a concerted national program with the highest level of science and standards, I think it is something that we are hoping with the new National Institute of One Health will emerge.
Thanks. Lots to do everywhere, that's for sure. Let me turn to the audience again. Are there any further questions or any comments from anyone in the audience? Yeah, in the back. What time is it?
Yeah, make sure you're not out of time.
The time has stopped here, so I think we can go.
Okay.
We can go on forever. Go ahead.
5 minutes.
Maybe
Okay.
you could start the timer again.
Real quick, so I think both panels previous to this talked about field research. How do you guys address field research in your own countries?
Okay, field research, anything else? Anybody else want to raise anything? We have 5 minutes, is that right? Anyone else? Yes, over here and in the back. Okay, fine, and then we'll come to you.
Well, I'm just adding to David's point. In infection prevention and control, there's the concept of universal precautions. Are universal precautions added in, are generally adopted in field epidemiology and sample collection?
Thanks. And then one more, and then we'll come to an end.
I really appreciate your frank discussions, particularly the way you phrase the problem of soft money and soft jobs. I think it affects most of us. So my question is, how do we solve it? Thank you.
Good question. So let me turn to Anurag first to tell us how he solves the question. How he solves the problem.
Solutions are tough.
And field research also, if you can say.
Let's take field research first. Field research is something that needs to be far more than is. I think one huge challenge in field research, preventing it from growing rapidly, is that what you may find may be unpleasant to know, and there is a desire to control how field research is done, how the data would flow. And I believe that's a universal problem. Whether universal precautions are observed, all the researchers I know would certainly do it. But I would temper that statement by saying I am quite certain that in a variety of animal-human interactions in the extremes of India, I'm certain the concept of trained precaution simply doesn't exist, whether handling animal carcasses or anything else like that. So I wouldn't worry so much about my field researchers. I would worry a lot more about other people who handle those things. I'll just give one brief answer to that one. And it's going to be an unpopular answer perhaps. The ideal biosafety lab should be balancing risk and benefit in such a way that essential work gets done. So which means you need to be doing fewer projects well. The competing way is doing many projects so that you can have the maximum overheads and everything else to keep the enterprise running. It's a conscious decision on the leadership part, what they want to allow. So for example, during SARS-CoV-2, many labs wanted to jump in and start culturing. It was a very conscious decision to only let the very best lab get into it, even though every organization around the world was probably saying, take the money and start culturing. So you have to resist it, and there is no other way for the scientists themselves and the scientific leadership with the scientific training to say this.
Nesreen, your views, and then I'm going to come back to Anurag and to you with
I
a
like
final statement of what you think is the most important part of this
It's
session.
hard to resist money.
So, Nesreen, go ahead.
Yeah, regarding field research, I will speak about my experience. In field research, we have, we're conducting a couple of projects in the field related to, bats and migratory birds. And we try as much as we can to fulfill all requirements regarding to safety and security, how to transport samples, and how to handle, different specimens from these types of animals. So in general, there is the point mentioned that everyone knows and are aware about, safety and security in the field and how to handle animals. The same point that there is no mechanism or there is no way in order to oversight these activities in the field. Regarding, precautionary measures, they are adopted by our epidemiologists in Jordan and I think, in most countries of the region.
Okay. Anurag, we're at the end. I want you to leave us with one message, one important message that you feel has come from these discussions.
I think we must realize that we work in a very critical area. It is most important to do things that are necessary and to do them well, and build the structures around our research that allow us to do it and not get distracted.
Thanks. And Nesreen, final word.
Maybe I will say that I'm really happy that I'm part of this project. I'm very privileged. Thank you for Professor Shahid, who introduced me to this project. It's really a good, event and a way in order to interact with different, experts from different parts of the world and to see their opinion in this domain. And thank you very much for this opportunity.
Thanks. And tomorrow we'll hear from both the African region and from the Latin American region on some of their problems in biosecurity and biosafety. So with that, I'll bring this panel to a close and just remind you that at 6 p.m. there are cocktails in the Glow Bar followed by dinner. So thanks to all of you for your attention and your questions, and thanks to the panelists for their answers.
Thank you, David. Thank you.
Thank you all.
Hey, c'est. Hey. Hey. Hey, bonjour à tous. Est-ce qu'on m'entend à cette distance? Forcément, quand les gars ils parlent à cette distance du micro et qu'ils sont endormis, ça ne fonctionne pas. Hey, c'est. Hey.
Après, justement, si c'est les Africains
Hey, c'est. Hey, hey. Ah, là, voilà, regarde, tu vois là le trim? Comme ça augmente. Là, hey, c'est. Bonjour à tous. Ah, si. Non, si, oui. Ah, allô. Hey, c'est. Hey, c'est. Hey, hey. Après, ouais, faut peut-être qu'il regarde un truc avec lui, hein, parce que moi je suis à plus 24 dB, son truc il est marrant. Après, quand gérais le gain d'entrée, ça marchait super. Hein. Ah, ça, pour le coup, c'était nickel. Ah bah quand j'augmentais le gain d'entrée, c'est pour ça, après il disait plus grand chose, parce que j'arrivais à... Enfin, en gros, j'ai augmenté le gain d'entrée pendant le truc. C'est pour ça que... tu fais jamais ça. Jamais de la vie, tu changes le gain. Quand ton gain il est calé, voilà. Voilà, c'est ça. Puis en gros.
Il y a quelqu'un qui reste pour la conf.
Moi, je suis là, ouais.
C'est toi qui
Ouais,
va montrer
ouais.
le manuel à Thierry.
Good evening. Good evening. How's everybody doing? This is so typical of a Bulletin event. Everybody is so engaged with one another. And being the after-dinner speaker is always a challenge. Good evening. I'm Dave Kuhlman. In my day job, I'm a senior partner at a firm called Lotus Blue Consulting, but it is my great privilege to be the chair of the governing board of the Bulletin of the Atomic Scientists. What a great day we've had today. It was a great discussion. I know I learned a lot. And it's a great start to the Conference for the Human Pathogens Project, and we're so grateful you're here. It is my honor tonight to introduce you to this evening's keynote speaker, Dr. Zeynep Tufekci. But before we do, I'd like to share with you a realization I had today, and it's actually a realization about the Bulletin. And it really was, I think, a realization about sort of what lies at the heart of what makes the Bulletin extraordinary. You know, at the Bulletin, we're the keepers of the doomsday clock, which is one of the great iconic images that galvanizes people's attention towards human-made existential threats. But I actually don't think that's the secret. We have an award-winning magazine, sorry, John Mecklin, where are you, with apologies, remarkable, but I don't think that's the secret. We have journalism that draws millions of people whenever there's a crisis in the world, and I don't think that's it. My realization this afternoon was that what lies at the heart of the Bulletin is a belief. And it's a belief that if you get knowledgeable, well-intentioned scientists and policy experts in a room, and you give them an opportunity to engage on the thorny and complicated issues that are associated, the blessings and the curses of modern science. That if you create an opportunity for them to engage both among themselves and with the public, that is our first best hope to secure our collective future. And so I don't want to go on without thanking all of you for coming and participating in that great effort, because that is really, in my view, the heart of what we do. And thank you in particular to Zeynep Tufekci, who is our keynote speaker this evening, and thank you for joining us tonight. Zeynep is the Craig Newmark Professor and Director of the Craig Newmark Center for Journalism, Ethics, and Security at the Columbia Journalism School. She writes regularly for the New York Times and the Atlantic. She is the author of Twitter and Tear Gas, The Power and Fragility of Networked Protest. She also has several TED Talks, which I would recommend. Her work focuses on social media, media ethics, and the social implications of new technologies, as well as societal challenges associated with the COVID-19 epidemic. In 2022, she was a Pulitzer Prize finalist for her columns on the pandemic and American culture, published in both the Atlantic and the New York Times. Zeynep has a way of presenting the issues and challenges we've discussed today in a manner that is both engaging and informative, and I look forward to hearing her remarks tonight. And I would also suggest to you that they represent an opportunity for us to explore the ways in which we might be able to characterize, package, and convey some of the ideas that are coming out in this conference tonight. So with that, Zeynep, over to you.
Thank you, everyone. I will first do the obligatory lower the microphone. It never fails. Second, I would like to thank the organizers for not putting me between people and their dinner, or people or their cocktails or dessert. That's sometimes what happens. You get to speak and people are waiting for you to get done so they can go have some food. I would like to thank them for not having that happen. I would also like to, I guess, thank, or not, let's see how you feel about it, the physicists who created the movement to try to warn about dangers of atom bombs, nuclear weapons, and all the need for the world to keep that under control, because that's the reason I'm not a physicist. I grew up in Turkey. I was one of those kids who was really into math and science, and like many kids who are into math and science, I thought I wanted to be a physicist because it sounded wonderful, unlocking secrets of the universe and finding all those things. And again, I was in Turkey, and this is a child speaking, and I only spoke Turkish, so I didn't really read all the history. I came upon what I would call the atom bomb problem that I think a lot of children who decide they're going to grow up being physicists encounter. And I thought, there's too much ethical burdens associated with being a physicist. This is so difficult. I don't think I want this. So I got into things like computer science or writing about pandemic and all the other things instead of physics. So that's a very, for me, interesting full circle to come to address this organization once again on a topic of great risk that comes from the great benefit that we also get from advances in science. Biology is not the first field, obviously. Physics was the first big one that came to light with how much science could help, heal, empower people, but also it came with real power. And one of the things I think about when I think about nuclear weapons is an essay by George Orwell that's not very well known, unlike his many other work, where he envisions the nuclear world. And he thinks that this will be the end of anti-colonial struggles because the big powers, US, Soviet Union back then, a few big European powers, they will have nuclear weapons and no colonial nation will be able to fight for their freedom because they will be nuked. He thought that's it. Now, we know that didn't happen. That did not happen. World War II, the post-World War II period, saw a huge amount of anti-colonial resistance and many countries becoming newly liberated from their colonial overlords. And one of the reasons that didn't happen, that was alluded to in parts of the panels, was that atom bombs are clearly not as easy to proliferate, so we know that part. But that doesn't answer why they weren't used. And in retrospect, if I was telling my child self about dangers of weapons, perhaps I would have thought about things like, the firebombing of Dresden and Tokyo, which were just as dangerous in their death toll as the atom bomb had been. But there was something different about the atom bomb. It was science, the fruits of science, the fruits of intellect, the best of us, having come up with an understanding of the secrets of the universe that ended up turning on ourselves under these conditions. And whatever else you may have thought about, in comparison to other horrors of World War II, it became clear that there was a massive, justified, understood revulsion against the idea of using the fruits of science for mass murder in that sense. And physicists led the way. Physicists who had worked on the atom bomb even joined this movement to say, we need to draw that line. And it became, so far, knock on some wood you find, it became unpalatable to use nuclear weapons. This whole tactical versus strategic versus this or that did not matter. The world that George Orwell envisioned, where the big powers would use smaller nukes to kill and terrorize anti-colonial struggles, did not happen. That was not just the Anti-Proliferation Treaty. That was a political line that got drawn by the public with the leadership of scientists and politicians, so they could not use it and remain in the family of nations, remain and retain the legitimacy in their own nation. I hope this does not change. This is not some law that's written in some stone that can't be changed. But that's what stopped it. It wasn't because there have been other wars, there has been mass murder, there have been all sorts of atrocities. So I don't have some, naive and optimistic view of it. But one existential danger so far has been kept in check through what the public has believed was an unacceptable use. So why does this apply to this question now? Well, partly because we had a pandemic, and the question of the origin of the pandemic has been completely wrapped into the biosafety question. Now, obviously, this is not the first pandemic we had. This is unlikely to be the last pandemic we had. And obviously, I don't need to tell a room full of many scientists that zoonotic spillovers are a common way in which pandemics spread or happen. This is historically not some unknown thing. But of course, we don't say we always had wars, therefore we don't care about nuclear weapons. We've always had pandemics, but we did not have the kind of biotechnology in labs we had until quite recently. So therefore, something has changed. We've always had wars, but once you have nuclear weapons as a tool of war, the concept of war fundamentally changes in what it represents as an existential risk. You have to also consider that. So, and unfortunately also, the question became wrapped up, at least in the United States, in the first year of the pandemic especially, with a president that wavered between declaring the pandemic not a thing, to the fault of China, to the fault of not a thing again, to declaring it might have been all sorts of theories that we know made no sense, demonized scientists in China, and correctly created a lot of revulsion among sane scientists that this was the discourse that was happening to a question that we did not have an answer to. And I believe that led to a situation in which that we have seen in the history of science before of what I would call overclaiming, overcertainty, and overreaction that did not acknowledge the basic fact that we do not know how this pandemic started. We may never know how this pandemic started. In pandemic investigations or any outbreak investigations are complicated, complex, even under the best of conditions, with full transparency and a lot of effort. Sometimes we don't know. Instead of acknowledging this, we've had a concerted effort by a small group of scientists and journalists who were gullible, and some politicians who wanted to shut off the crazies and the lunacy, which I completely sympathize with, to a wave of overclaiming that we had an answer. And of course, again and again, it turned out we didn't have the answer. So we had waves of media where the media would go to bat and say, we finally have an answer. And then, of course, it would turn out we didn't have the answer, and the journalists were just being gullible to their sources. We had it with claims of dispositive evidence in preprints that didn't turn out to be dispositive evidence, and they weren't and couldn't have been. But the damage was done because journalists echoed these without being critical enough. And as a consequence of this, we are seeing an increasing percent of the global public, the US public, the American public, believe overwhelmingly that there is some knowledge that it did start from a lab. In fact, the numbers are so lopsided that it's kind of really unnerving. 60 to 70% of the US public, depending on the poll, believes we know it was a lab, whereas only 20% thinks there was no research involvement. There's a small group of people, down to 11%, who, like me, believe, well, we don't know. So what has happened is this effort to claim the mantle of science without the evidence to support that, and journalists going along with it, and some possibly well-meaning politicians going along with this, has created this very explosive situation where a large majority of the global public believes that this pandemic started in some lab or research incident, that they don't know the details. Now, you might be tempted to believe, well, the public believes creationism and all of that, and I hear that all the time. It is not that, because the science, the sort of belief in science has gone up. The idea that it might have started in a lab is because scientists have lost credibility on this question by 2 years of overclaiming, 3 years of overclaiming, claiming they were the science, censorship by social media companies and other media companies, newspaper articles and all sorts of major news articles, including very recently that made claims that did not turn out to be true or supported by the evidence. And even if you think that you're of the opinion that it's likely no research relationship, that's a fair opinion to have. People can have opinions. This is very, very dangerous for science. This is very dangerous for credibility of science, to have so much of the public believe that scientists are lying to them and covering up to them. And the idea that this is just the same kind of people who believe in creationists is just not true. I'm an academic. I talk to academics with PhDs and all sorts of high degrees who don't follow the few loud people on Twitter or don't really read the few newspaper articles. They're completely convinced the cover-up is deliberate because they're not stupid. They can see that there's been a concerted effort to deny one possibility as unscientific while pretending the other one has been proven. So they get suspicious. And then they start thinking, why are they doing this? And they incorrectly, in my view, conclude that's because it's a deliberate cover-up of a known fact rather than, no, it's because of the polarization in the first year and a few loud scientists circling the wagons and journalists parroting them rather than doing their job. And a lot of people being intimidated in the fields to speak out. Like, I think that is the truth. I don't think that there's a bunch of people who know something who are covering it up, but that's what it looks like from the outside. That's what it looks like to a lot of smart people. This is not just a bunch of creationists who believe anything. And you can see it in the poll numbers. And I think it would be dangerous for people to kind of assuage themselves that the poll numbers represent gullibility rather than distrust. And the pandemic has had a lot of issues with public health and science. And again, science increases trust, but not on this. This is very important. So this is the perception part of it. And I think it's very important for sane, sensible, logical scientists who are not of the let's say the loony crowd, the ones that are trying to get Anthony Fauci killed and the ones that sort of make it so that Ralph Barrett can't safely go to a restaurant, the ones that I oppose wholeheartedly. These people in this room, scientists everywhere, should take the microphone away from the people who are making those kinds of claims and who are being very unscientific and illogical and dangerous, in fact, to science. But they're thriving because of this vacuum. If one side is overclaiming and saying, we represent science, everybody else shut up, and if the people who kind of correctly say, look, we don't know, and the fact that it was plausible that we're wondering about this is enough to consider biosafety to be a major question, along with, of course, worrying about spillover risk from other situations. If we don't take up that space will be taken up by people who are saying things that are not scientific, that are not sane, and that are driven by political polarization. But if there's nobody else in the space, that's who speaks. And that's why I wanted to come here. When they invited me to speak here, I said I have to speak because now, finally, a bunch of people who are concerned about biosafety, who are trying to say something, and who are taking it seriously, because it needs to be taken seriously. Now, in 1977, we had a H1N1 pandemic that was triggered by almost certainly a vaccine trial that had gone wrong. What had happened was there was some fears of another H1N1 like the 1918 pandemic, and scientists very understandably started doing vaccine trials. And vaccine technology back then was less safe, to say the least. There was a lot more potential for, let's say, escaping the safeguards. And it turns out that did happen. And then we had this pandemic that had very weird features. Okay, so we didn't know what had happened. The features were, it didn't affect people under over 26 as much. Why that age? And then you look at the genome, which was a newer technology back then, right? Now not as accessible as today. It's the same as the one 26, 27 years ago. That's not how evolution works. And there were some further tests on its sensitivity to temperature and things. And scientists quickly realized this is a vaccine gone wrong. But there was a 20 to 30 year silence that was deliberate. And it's been written about as scientists deciding at the time that they did not want to come out and say, yeah, a vaccine trial gone wrong caused a pandemic, because they were afraid that it would just damage the nascent cooperation on flu with Russia and China, back then Soviet Union and Communist China. So they kept quiet. And the topic just didn't come up much for 20-some years. And finally, one of the sort of very senior scientists said, yes, my counterpart wrote to me that it was a vaccine trial gone wrong. So we got it wasn't a secret, but it wasn't talked about on purpose. You have a similar crisis in the 1989, the Sverdlovsk anthrax. That one is genuine lab leak. They forgot to change the filter, and it was a genuine bioweapon research, and then it leaked into the area, and then killed people and sickened hundreds. And it was, of course, covered up because it's the Soviet Union. And then some very high-level scientists, a couple from Harvard, very, very good people, they went there and concluded they bought the official story that it was meat poisoning, despite the fact that it didn't fit anything. And one of the things that really struck me about that case was the scientist in question who vouched for the Soviet Union. He was a very, very good scientist. He was not some gullible person. And he had been at the forefront of trying to stop the Reagan administration from going into bioweapons and weapons research. And some others have speculated at the time whether not a deliberate lie, but a motivated reasoning to not want to admit that this did not look like mass meat poisoning. In fact, next year, a Wall Street journalist, not a scientist, but a Wall Street journalist went to the same place, started digging, and immediately became clear that the official story had no legs. And it was, in fact, an anthrax outbreak from a lab. So it just took a journalist who was not going to be gullible and motivated to find out once he was able to do research. And this is at the time the Soviet Union's kind of collapsing, so all of a sudden you had the thing. So we've had incidents in the past where scientists have, I'm not saying insincere beliefs, but sincere beliefs that color how they think about, how they communicate about risks from their own field, which is very human, very understandable, but to the degree it may have worked to some level in the past, it absolutely backfires in the wrong direction now. It absolutely backfires. It threatens the credibility of science. If we had gone with what I thought was always the same thing to say, which is that, look, we don't know how this pandemic started. Yes, it started, the first known outbreak is in a wet market, but there's also a lab that deals with bats just a couple hundred yards from it. Who knows? A field researcher got infected and went to the market. They had live bats. They had bat samples. Bat samples can get pool and give rise to recombinations. So that you might get infected. If you have bats, you might get infected by things you didn't even know they had. You don't have to be researching coronaviruses to get coronavirus from a live bat or a bunch of bat samples that recombine. So you could say all of these are possible because it really is weird that it happened in the city. But on the other hand, we also have a wet market, so it's absolutely possible that there was some animal that was infected that came to the wet market and infected the human. It's also possible, like the way we have infected minks with the coronavirus pandemic. In Denmark, there was huge outbreaks of coronavirus among the COVID-19 among the minks, and they generated their own new variants, which then infected humans. Now, imagine if you didn't know the backstory. It would have looked like a mink origin, whereas, in fact, a person had first introduced them, but because minks are kept in a cramped condition and they have upper respiratory structures that are similar to humans, they generated variants and infected humans back. So it didn't originate from minks, but it would have looked like it if you had started the date, from the mink infection on. So it's possible that there was an animal outbreak, but that the intermediary animal was a human. There's nothing that says that this couldn't have been a bat-to-human direct infection. The intermediary animal is not some scientific requirement. In fact, a lot of scientists, including Ralph Barrick, who studies coronaviruses, thinks we might have infected civets rather than civets being the intermediary. It's also possible. Pandemics are rare events. They chain together rare events. You cannot do regular probability. A villager from Yunnan might have traveled to Wuhan that week in November, whenever it started, and started the outbreak. Like, there's no way to rule out something really rare happening because spillovers happen all the time, pandemics do not. We have spillovers all the time. So which brings to my final question. So what do we take away from this? I think one thing we take away is that we should stop overclaiming. It is hurting science. It is hurting the credibility of science. It is making the public very, very suspicious of scientific research in this area. It is not good. The second thing is we should acknowledge the risks are real, and saying, oh, pandemics happened before sounds as stupid as it sounds because we did not have this technology before. When people go on TV and say it must be spillover because we always had pandemics, they do not sound convincing. Everybody knows we didn't have the technology before. Like, that doesn't make sense. So those are arguments we should not make. We should acknowledge that biotechnology has advanced to the degree we can do all sorts of things, some of which are potentially dangerous. So that means we have to think really carefully about the ones that could potentially trigger a pandemic, because that is not a minor risk. With SARS, too, this one, it escaped from Taiwan, BSL-3 lab. The researcher got infected. She kept it quiet, and they finally found it, by testing. And if it had been a new virus, we might have never known, because the only reason we know it happened is Taiwan's very transparent, very good for them. But the only reason we know this is because we caught it, because Taiwan was zero COVID, was testing like crazy, so could pinpoint what had happened. If she had been working with a pandemic potential pathogen, we never would have known, because, how are you going to know? You don't already have the test. And we know that a lot of these pandemic potential pathogens were being worked on BSL-2 and BSL-3 at best. So we have a real-life example of exactly how it could have happened. Is it proof? It is not. Do we have to acknowledge that it could have been? Yes, we need to. So what you do going forward, in my view, is we go back to what we should have said in the first place and say, yes, this technology is powerful, the science is powerful. It's great in some ways, we get a lot out of it. So we have to figure out what of the things we do are worth the scientific merit. So for example, should we really be poking around all the bat caves to try to figure out all the potential? I'm not sure. Like, there's a lot of argument that we should just surveil people for early outbreaks rather than introduce an extra risk by poking all the bats to try to collect their samples, because in Wuhan, as far as I know, there's only a couple of places with bats, and those are all labs. It's not any of the markets. So it's an unnecessary level of risk. The second thing is, in the phrase, spillovers happen all the time, but pandemics do not. Pandemics seem to require, for very obvious reasons, density. You do not want the highest risk work in big cities. I'm not sure how this is so unclear yet, but it is. And I actually argued and had discussions and interviewed dozens of virologists on this topic. Almost all of them were very adamant that it couldn't have been a lab, even though I thought, like, you don't know. But some were willing to concede they couldn't rule it out. And I asked every one of them, I said, shouldn't some of the high-risk things, if you think there's scientific merit, shouldn't they be moved out of cities? Like, I'll buy the argument that you're doing something that is super essential. Why is your lab worker going home at night? Like, because we know from research, we know from investigations, most lab-acquired infections are not detected until testing. So if you don't know what you're testing for, you're not going to detect it. This is such a clear history. Lab accidents that infect people do not get detected until testing most of the time. Like, this is sort of this hard fact. So why is your lab worker going home at night? If it's that important, surely we can build labs of the highest quality somewhere, and you guys go spend a month in it. And everybody said, no, 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 no. Now, I'm an academic. I live in New York. I understand. I do not want to move a month somewhere to do my IRB application either. Like, I get it. But maybe that would also make people think, am I doing this for the scientific merit, or am I trying to get a Nature Science Cell paper because they'll publish it? Right? If you had to do some extra work to prove that this is such essential work, perhaps it would align the incentives. Alison Young has a new book coming out. I haven't read it yet. It's coming out in a day or two. And one of the first things she revealed is that there was a lab incident with H5N1 that was engineered in Wisconsin, that University of Wisconsin, that the University of Wisconsin had promised they would alert public health authorities, and they did not. This is the United States. I know we like to point our fingers at China or this or that, but like in the United States, an engineered H5N1 lab accident where the hose disconnected did not get reported to the public health authorities despite promising to do so, and they let the worker quarantine at home, I believe. And if we know anything about, there's studies from the pandemic airborne transmission where like literally a 5-minute same room across 30 feet, I believe there's one in Korea, transmission. Like, this is not the kind of risk you take with an engineered H5N1, and yet they did. They didn't tell anyone till later. If US universities are doing this, you can imagine in a place where the stakes are higher or more authoritarian, it might even be worse. We are this little away from an incident, hopefully not a pandemic, an outbreak, that is going to turn the entire public against all of virology. Right now, it's already 60%, 70%, very suspicious. One more incident where they believe this is not being disclosed properly, it's going to genuinely damage our ability to do the kind of science we should be doing, rather than what we should be doing now, which is reconsider what are we doing, what are the sort of safety standards we need to be doing, and how do we get, and this is sort of my plea to the people in the field. There are very few of them who have been outspoken. They have been attacked occasionally by their colleagues. There's a lot of circling the wagons and tribalism going on, and I understand. This is why I didn't want to become a physicist. It felt very heavy to consider such ethical things. But here we are. But it was the people who stood up, the people who spoke out, that ended up being the best face of that science to the world. The people circling the wagons ended up discrediting their own science, even if in the short term they can convince a journalist or two, or maybe win some Twitter tweets or whatever virality. In the long run, they damaged their own field. And it was the ones that stood up and spoke for science with credibility, with humility on what the risks were, and try to sort of say, how can we do this safer to benefit people while also curtailing these risks? Those are the ones that keep the credibility of the science going forward. So that's kind of my plea to the field to the degree we can make it, and a thank you to those few of you, some of you are in this room, who have managed to speak out despite all the pressure to conform and to sort of go along with the circling the wagons crowd. If I think it would have been a great loss if everybody had done so. And in the meantime, I think institutions like this and a bunch of you work in these institutions, please, please, please take up that space that is now being occupied by people who are overclaiming with certainty, with a sense of tribalism, perhaps sincerely in the name of their science, but not really doing something in the long run. And they are empowering the very voices, the unscientific voices, the ones that are just motivated by polarization, that see bioweapons under every stone, like the ones that you see on Twitter, the ones you sometimes see on congressional hearings. That's what's empowering them. The fact that other people aren't speaking out, the legitimate institutions aren't stepping in. I applaud the WHO for refusing to say this is over and we're going to, not think about this anymore. They've been very clear that the question remains open. I applaud sort of the scientists who continue to do that. And again, the very thing that makes science so wonderful and powerful and so amazing and a miracle of sorts, I guess, to the degree you want to call it a miracle, I would call it a miracle, is the same thing that gives us this edge and this power, and not grappling with it does not make that edge go away. It just makes the public really afraid of it, and they have a point. And thank you.
And for the rebuttal, no kidding. Thank you, Zeynep. Two quick housekeeping items. Number one, we will start tomorrow 9 a.m. in the WI room, same room as before. Secondly, I'm told, and I'm going to read this exactly, please make sure to bring your name badge. Must have name badges. Now, it does not say what the consequences of not showing up with your name badge are, but let's not find out. And we'll see you tomorrow morning at 9 a.m. Thank you.